Hello my dear friends, welcome to Live Prep. Myself Dr. Gajapati Verma. As I informed you earlier, under the name of Bootcamp, we are providing both revision lectures and also test series. Revision lectures are available both on Live Prep app and also on YouTube channel. And then this particular test series will be available only on the app. Clear? And now let us quickly go with the revision. So in this lecture, we are revising cell cycle and cell division chapter. This chapter is comparatively smaller one, so that's why in single lecture we will try to complete this one. But the important thing is, though it is a smaller chapter, the weightage is very high and it is a highly conceptual one. Many students are trying to by heart the events of cell cycle and the content. Don't do that, try to understand. So that is the reason why once we are going with NCERT content, at the end of the lecture, so I will try to make one table. In that table we will discuss, so what is a diagram for a particular phase and what is the ploidy, what is the DNA content, what is the type of chromosome, names and everything. So all these things we need to observe very carefully. So that's why at the end of the chapter we are doing that, but the basic names that are required I will cover in advance, so that at the end of the lecture, so you can easily understand all these things. One more suggestion is, don't try to by heart these contents. Try to understand what exactly happens after one phase. So first, for a particular phase you are observing the diagram. So what exactly happens in the next phase? What exactly happens after that? So in a sequential manner you need to observe it, understand it. So then this chapter will be very easy. Clear? And not only this, there are some concepts that are interlinked with some other chapters. So we need to observe that interlinking also. So all the things we will observe in this particular revision lecture. So that's why this lecture will be little bit lengthier. I am telling you in advance itself. Okay. So but entire content we need to cover in single lecture. So now let us start with the revision of cell cycle and cell division. And before directly going into the chapter, first we will go with the unit introduction. But if you are clearly observing this unit introduction, they have given some sentences related to cell, the unit of life, and some sentences related to biomolecules. But they have not given any sentences related to cell cycle, cell division. So we don't need to worry about this unit introduction. So now we can directly go with the chapter. So here, first what they are saying is, they are giving some general introduction. What they are saying, even the largest organism should also start its life from a single cell. What is that single cell in this case? It should be the zygote. Okay, so this is what the information they are saying. Next one, growth and reproduction are characteristics of the cells, indeed of all living organisms. There are some exceptions to this particular sentence, and these sentences you will study under the chapter called as living world. So here I don't want to tell all those things. So here we are simply following this particular sentence. So cells, they are able to grow in size and they are able to divide. So that reproduction is nothing but, reproduction with respect to cells is nothing but what? It is a cell division. So in this way, organisms are also able to grow and reproduce. Cells are also able to grow and reproduce. So try to understand the basic information. So this is what grow means. Here the growth means increase in size. Reproduction means the number will increase. So this is what the basic meaning here. Okay. Next one. So here in the case of this cell division, generally we are talking about parental cell divided into two daughter cells. Please observe one. This is a general and very important sentence. Why only two cells? We are actually studying about two types of cell divisions. One is mitosis, other one is meiosis. Understood or not? One is mitosis, other one is meiosis. In the mitosis, parent cell will divide into two daughter cells. In the meiosis, parent cell will divide into four daughter cells. Clear? But in the NCERT, what they are saying, in the case of this cell cycle, so parent cell will divide into two daughter cells. Why they gave only two daughter cells? Is the sentence true always? So you need to observe about this one. So now you need to understand the basic concept. So what is this? In the case of mitosis, two daughter cells are produced. In the case of meiosis, four daughter cells are produced. Correct. But here, whenever you are using a term called as a cell cycle, cyclical event should occur. So for which one you are using the term cell cycle? The term cell cycle is used to only, I am giving you options, only mitosis, only meiosis, both. Now you just observe it. Many students are thinking that cell cycle term is used for both. This is what many students are thinking, but it is not true. Cell cycle term is useful or used only for mitosis. you should not use for meiosis. Why is it like this? Observe carefully. So anyhow, you know all the concepts. So quickly I am revising this. In the case of mitosis, what you are saying, we are starting with a single cell. So that is in G1 phase, then it is going into S phase, then it is going into G2 phase, then it is going into M phase. At the end of the M phase again G1 will come, but cells how many will be produced? 2 cells are produced. And again these cells are again from G1 they are going into S, G2, M, and again they are coming. So in this way what is happening now, here there is a cyclical events, cyclical events, cyclical events, cyclical events. So in this way cyclical events are occurring only in the case of mitosis. But what about the meiosis? In the case of meiosis, if you are observing, suppose a cell that is present in G1 phase, now it is entering into S phase. Read the NCERT carefully, they are not talking about G2 phase in meiosis. So we are not writing G2. Directly it is going into meiosis I and then meiosis II, and at the end of this 4 daughter cells are produced. That's it. And will these cells again enter into these phases? No, they are not entering into these phases. So that's why cyclical division concept is not applicable to meiosis. Cyclical division concept is applicable only for mitosis. So that's why cell cycle, the term cell cycle is applicable only for mitosis. Please observe carefully. This is what the reason also you should know, right? So if you don't know the reason, then there is no use in studying all these things. So technically cell cycle word is useful only for mitosis. So with respect to mitosis, what you need to say? Parent cell will divide into 2 daughter cells. That's why. This is what the logic behind this particular sentence. Understood or not? So that's why generally whenever we are studying about the cell cycle, always they are talking only with respect to mitosis, because this is what the reason is. So understand the reason behind this. Next one. Generally in the case of cell division, so our cell cycle, what we are saying, so DNA replication is observed. So DNA will replicate, whether it is mitosis, meiosis, whatever it is, DNA replication is occurring, and then cell is growing in size, and then it will undergo division. So in this way DNA replication, cell growth, and then division are taking place in mitosis and meiosis process. Okay, so this is what they are saying. Next one. So in the case of cell cycle, what actually the cell cycle, there are some phases or the events that are taking place, and again in each particular phase and different things are taking place. So under the heading of mitosis we will study individually, under the heading of meiosis we will study individually. Okay, so in this way, in the case of cell cycle, different phases are present, and in each phase again different events are taking place. Clear? And all these things are happening in a coordinated manner. So an entire this particular process is called as cell cycle. Clear? That's it. Next one. So here exactly what is happening in general, if you are talking what exactly happening in the cell division. So cell growth with respect to cytoplasmic increase in size is taking place. This is what they are saying, and this is a continuous process. What is the meaning of this? In the G1 also cell is growing, S also cell is growing, G2 also cell is growing. Remember this sentence. When we study about G2 phase, I will add one more point. Okay, cell growth in terms of cytoplasmic increase is a continuous process, but in which phase exactly maximum growth occurs? Question is very clear. Growth is a continuous process. In all the phases it is growing, but out of these phases, in which phase exactly maximum growth is taking place? I will link up this information with a sentence that was given in the summary. I will link up when we are studying the individual events. So remember this particular question. Next one. DNA synthesis is occurring only in a particular phase. What is that phase? S phase, synthesis phase. Okay. And next one. So in the case of S phase, what is happening? DNA replication occurring. DNA replication means what? Chromosomal duplication. That means hysterochromatids are formed in the S phase. And this replicated chromosomes or replicated DNA is distributed into daughter nuclei. By the end of this particular M phase, so telophase will be observed. So in the telophase, now this DNA is distributed into 2 daughter nuclei. The daughter nuclear term we are using in the telophase. Yes or no? There. After that cytokinesis will occur, so that the DNA replicated in the S phase will be distributed into daughter cells after cytokinesis. Yes or no? This is what finally happening in the case of cell cycle and cell division. This is what they are saying. And all these events are under the genetic control. Understand this sentence. You need to link up this sentence with human health and diseases. In the human health and diseases, we are studying about the cancers. And there also we are studying about the proto-oncogenes. Proto-oncogenes. Proto-oncogenes are the normal genes that are controlling all the events of cell cycle. This is what. Under genetic control, they are saying, no, what are the genes that are controlling? They are called proto-oncogenes. If proto-oncogenes are mutated, they are converted into oncogenes. So oncogenes will cause the cancer. So that's why what we are saying, normal genes are proto-oncogenes. They are controlling the cell cycle events. That's why cell cycle is happening normally. If they are mutated, now cell cycle regulation will be lost. If cell cycle regulation is lost, what happens? Uncontrolled division will occur. Uncontrolled proliferation will occur. So that's why it will lead to formation of mass of cells called as a tumor, and that may be benign or malignant. Malignant tumors are called as cancers. So this is what the relation between these sentences. You need to interlink with various chapters also. That's why don't by heart these sentences. Try to understand the meaning and try to interlink with various chapters so that your concept will become strong. Clear? With this, the basic information is completed. But why these cells are undergoing the division? This information was given under the significance of mitosis, but I am discussing in advance. Now, what is this particular concept? Why the cell is trying to undergo division? Now, here you need to understand a particular concept called as nucleocytoplasmic ratio. Nucleocytoplasmic ratio, and it is represented with N by C. Very good. Up to this it is correct. But what is the mistake done by not just students, even some teachers are also making mistake. So what the mistake I will tell you, then I will tell you original concept. What is the mistake? We are writing N by C, we are writing. Up to this it is correct. But what some teachers are doing is, they are writing this N as N, 2N, 4N. That means they are writing this as a ploidy. They are writing this one as ploidy. They are writing this one as a ploidy. And this C they are writing as C, 2C, 4C. That means it is the DNA amount. DNA amount or DNA content they are writing. They are writing like this. They are writing like this. And please remember, this is a blunder mistake. This is a blunder mistake. They are saying this one N by C. They are writing like this. And this is a direct blunder mistake. Please don't do this particular mistake. This is a separate concept. This is a separate concept. Don't link up with this concept with N by C. Don't do mistakes. The thing is, the information is very clear and simple. Unnecessarily, teachers and students, they are making it complicated and they are making ultimately wrong interpretations. So don't do wrong interpretations. So observe this carefully. What is this N by C? N is nucleus and this C is cytoplasm. N by C is nucleus to cytoplasmic ratio. So nucleocytoplasmic ratio you are writing. Up to this also it is correct. Now 90% students know that it is nucleocytoplasmic ratio. But which ratio? Which ratio you are talking about? Is it area ratio? Is it surface area ratio or the volume ratio? This point also you need to know. So actually it is the volume ratio. It is the volume of nucleus to volume of cytoplasm. This is the point. This point you need to understand. Now what is this volume of nucleus to volume of cytoplasm ratio? What exactly it gives? So now observe carefully. Whenever a cell is newly formed, generally its size is small, and here it is having a nucleus. Sorry, it is having a nucleus. So now if you are observing, this is the nucleus and this is the cytoplasm. Yes or no? For the newly formed cell, what happens? Now here it is having a particular value of N by C. Some value is there. Now what happens? When the cell grows, when the cell is increasing in size, now what happens? The size of the nucleus increases. Size of the nucleus increases. That means volume of the nucleus increases. Though it is increasing, the nucleus volume increase is a little bit less. But what happens to the cytoplasmic increase? The volume of cytoplasm is increasing enormously. Please observe carefully. Nucleus volume is increasing. Cytoplasmic volume is also increasing. But what is the rate of increase here? The nucleus volume is increasing, but this increase is very, very less. But if we are observing the cytoplasmic volume increase, that is very, very huge. And now you just observe carefully, N by C value. Nucleus volume increase is very, very less, but cytoplasmic volume increase is very, very huge. So now when C value increases, what happens? Now here you are getting some value, and this value is definitely less than this particular value. Yes or no? Yes or no? So in this way, for the smaller cells, you are having a particular value of nucleocytoplasmic ratio. But when the cell grows, that means when it increases in size, what happens to the volume value of this N by C? N by C value decreases. This is a very, very important point. Many students are not understanding this concept. When cell is increasing in size, cytoplasmic volume increases. That's why N by C value decreases. N by C value decreases. What is the original meaning of this? This is the mathematical level. At the mathematical level, you are doing this calculation. But in simple terms, if you want to understand what is the conclusion of this, observe carefully. Here the cell is small and the nucleus can easily control all the activities of the cell. When N by C value is more, so here I am saying more, here I am saying less. When N by C value is more, what is the meaning of this? Nucleus has more control over the cell. But here what happens when the cell is increasing in size? Now the nucleus, through the DNA, nucleus has to control all the cellular activities. That's why nucleus is called as a brain of the cell. So nucleus has to control all the activities, but the cell size is increasing. So many reactions are occurring for the maintenance. So now if I am using a word called if the size is keep on increasing, now after a particular point, what happens? The volume is very huge, so nucleus may not be able to control all the activities. If it is keep on increasing, the nucleus may not be able to control all the activities, and that is dangerous for the cell. So that's why after a particular growth, the cell has to divide again to form smaller cells. That's why smaller cells will have the greater control. So in the case of smaller cells, nucleus will have the greater control, and that is actually required. So if the cell is keep on growing up to a particular size, yes, nucleus can easily control. So that's why up to this particular level only the cell will grow, and further growth will not occur. So when it is trying to grow further, then it will divide. So this is what the main conclusion is. So that's why generally what we say, what is the overall conclusion here? The cells will divide to maintain a particular nucleocytoplasmic ratio. So that's why in NCERT they used a word called as to restore original nucleocytoplasmic ratio. So this is the normal nucleocytoplasmic ratio. To restore this, to get back this, large cells have to divide. This is what the original concept is. This is the reason why cells are dividing. Is it clear? This is what the point is. So smaller cells, now here the value. So here see this. So here nucleus volume is more and cytoplasmic volume is less. So that's why N by C value is more. And when nucleus volume is less, cytoplasmic volume is more, N by C value is less. So now where you can get greatest control? Here you can expect greatest control of the nucleus over the cellular activities. But here it may have less control. This is what the overall concept of this. So try to understand the concept. So that's why cells are undergoing the division. Now you understood why cells have to divide. Clear. Next one. So here, do all the types of cells have the same timing for the cell division? No. Different organisms, even in single organism also, in different locations, according to the functions, the cells will have different timings. It is called a generation time for the particular division. Okay, so the time that is required for division of one cell into two cells, we are talking with respect to mitosis. So one cell into two cells, that time is called as a generation time for that particular cell. Please remember, even in single organism, let us take the human. Do you think that in the human all the cells are having same generation time? No. If you are talking about the human skin cell, here we are taking the example of human skin cell. Human skin cell is approximately taking 24 hours. Yes or no? Suppose you are talking about the stomach. So in the stomach also there is a lining, and those cells, because of the HCl activity, these cells are continuously damaged. So there The cells have to be replaced very quickly. So that's why even in single organism also in different locations cells will have different generation times. But for study purpose we need to take one. Then here we are taking the example of human skin cell as an example, and there it is taking 24 hours time. Clear? Next one, regarding the yeast, if you are talking, approximately it is taking the time of 90 minutes, and these two are eukaryotes. And in the NCERT, they are not talking about E. coli or any bacteria. They are not talking about E. coli or any bacteria in the cell cycle cell division chapter. Why they are not talking about any prokaryote? This is a very important logical question. Think and tell me the answer. Why in NCERT textbook they are not talking about any prokaryotic division under the heading of cell cycle cell division? Can you guess? What is the logic here? The simple answer is cell cycle concept. In the cell cycle concept, what we are studying? G1 phase, S phase, G2 phase, M phase, all these things we are studying. And these phases are not applicable to prokaryotes. Please understand this. Prokaryotes, they are dividing by fission. In the bacterial fission, if you are taking example, in the bacterial fission, do you find all these phases? No. In the bacterial fission, only two phases are there. One is DNA replication, and the DNA replication is followed by bacterial fission. That's it, only two things. And immediately after that again, in the new daughter cell, DNA replication will start, and that will be followed by bacterial fission. Only two things are there. So, in the case of prokaryotes, these cell cycle phases concept is not applicable. Please understand this. That's why in the NCERT, they are not talking about prokaryotes. In the NCERT, always you are talking about only eukaryotes under the heading of cell cycle cell division. This is the important point. This point also you need to notice. But anyhow, for comparison purpose, I am talking about the E. coli. So these are the eukaryotic cells having different time points, different generation times. And here we are talking about the E. coli. And here already in the molecular basis of inheritance, under the experiment of Meiosis and Stall experiment, we studied that the generation time for E. coli is 20 minutes. And this point already I have mentioned under the cell chapter revision lecture part 1. There we studied that prokaryotic cells will divide rapidly. Yes or no? Multiply more rapidly than eukaryotic cells. E. coli is a prokaryotic cell. Within 20 minutes it is able to divide. But eukaryotic cells, they are taking more time. That means prokaryotic cells are dividing more rapidly. And when we studied about that sentence, I have linked this information. So link up the informations. Don't try to by heart this. Link up so that you can remember for longer duration. Clear? Next one. Before directly talking about this particular mitosis and meiosis, first I will tell you this, what is the ploidy concept and what is the C-value, because within the NCERT, within one orange color box, they have given some information. In the onion root tip cell, so and so number of chromosomes are present, suppose, and then in a particular so and so phase, what is the ploidy, what is the DNA content, likewise some questions within a box they have given this information. So that's why every student should know what is the ploidy, how to identify the ploidy, and what is C-value, how to identify C-value, and in a particular phase of a particular division, maybe mitosis or meiosis, what is the exact ploidy, what is the exact C-value, that you need to know. So that's why at the end of this particular lecture, I will make a separated table, I will draw the diagram, I will write the phase name, I will draw the diagram and what is the ploidy, what is the C-value, what is the type of chromosome. And if you know that particular table, you can write any question from this particular chapter. Okay, so that's why before going into this particular mitosis, first let me explain the ploidy and C-value very quickly so that it is useful at the end of this particular lecture. First, we will talk about the ploidy. What is this ploidy actually? Ploidy generally we are representing with N, small n. N means it is haploid. Anyhow, very clearly I have given this particular explanation in the regular lecture. So that's why I am directly going with the revision of this particular one. N is the haploid. 2N is diploid. 3N is triploid. And then 4N is tetraploid. Likewise, we are using the names. But in the case of these particular mitosis, meiosis, in general, for the cell cycle cell division, we are writing only these three. 3N we are not studying anywhere. Haploid you will study, diploid you will study, tetraploid you will study. Only these three things you are studying under the cell cycle cell division. So my focus is also on these particular three names only. Next one. Regarding the C-value, what actually the C-value is? It is about the DNA content. DNA content. It is also called as amount of DNA. Amount of DNA or DNA content. Any of these names they may use. And here also we are using C. This is actually capital C. Capital C. C means it is one particular amount. Okay, this is the amount. I will give the definitions with examples, diagrams. Don't worry. And then 2C, 4C. Here also we are using only these three. So with respect to ploidy, N, 2N, 4N. With respect to C value, C. C means 1C. You can write 1C or C. Okay, no problem. 2C, 4C. Only these three we are studying. Okay, now I will give the diagrams. With the help of diagrams, I will explain. And these diagrams also you will definitely encounter in either mitosis or meiosis. So whatever the information I am saying, entire this information is useful to understand mitosis, meiosis with diagrams. So at the end of this lecture, when we are making diagrams, this content is useful. Now observe carefully. So in the case of interphase, the DNA or chromatin thread-like structures are present. Yes or no? Like this. Or so in the case of meiosis, so you are observing this condensed. But here in general, if you are talking, here how many chromatids are there? Single chromatid. Here also it is single chromatid, but it is uncondensed. It is decondensed, thread-like. But the same thing when it is condensed, it will be like this. Either like this or like this. It is present. And other one is this one only, thread-like structure only, but it is dyad, right? This is monad. Single chromatid is present, that's why it is a monad. Here two chromatids are present, it is called a dyad, dyad. Many students are writing this spelling as a DIAD. It is not DIAD, it is DYAD. So observe the spellings also. Next one, it may be like this, like decondensed form, or the same thing if it is condensed, you will draw like this. Okay, this is what. And now these are the same diagrams, but how many are present? That is also important. Now what we need to do, in different species, the number is different. But what we are doing is, I will explain you with respect to human, because you can easily understand for the human. In the human, in the haploid condition, 23 chromosomes are present. In diploid condition, 46 chromosomes are present. So now let us see here. If they are present like this 23, what is the meaning of this? Single thread-like structures 23 are condensed, but this is also single chromatid, like this 23, like this 23 or like this 23. Condition will be same only. Okay, clear. Now like this thread-like structure, double thread-like structures. Okay, so now it is a dyad, whether it is thread-like or condensed chromosome, whatever it is, if 23 are present. Clear. Don't think that some are like this, some are like, it is not like this. If all of them will be having single form, they all the 23 will be like this, or all the 23 will be like this only you are talking. Now with respect to 43, so once again draw the diagram. So like this or like this. How many? If 46 are present. Next one, thread-like structures like this or like this. Okay, please remember this is the centromere. This is the centromere for all these things. Like this 46. Please observe whether it is a mitosis or meiosis, only these types you will find. Only these four. One of these four will be observed. One of these four will be observed in the mitosis and meiosis diagrams. That's why if you understand this particular information and if you know the phase diagram, you can easily write the ploidy and C values. Okay. Now we will go with the ploidy. So now if this is the condition, that means in a single cell, this is the condition. Then what is the ploidy? What is the ploidy? First we will write the ploidy. How you will write the ploidy? Ploidy is with respect to number of chromosomes. How you will count the number of chromosomes? How you will count the number of chromosomes? It is based on number of centromeres. Please observe this. Centromeres concept is very important. Okay. Please remember here it is called as one chromosome. This is also called one chromosome. This is also one chromosome. This is also one chromosome. Why it is like this? Here it is one chromosome and having one chromatid. This is also one chromosome but having two chromatids. This is the centromere. This side is one chromatid. This side is other chromatid. And the question here is, why you are considering this one as one chromosome and this one also one chromosome? Why you are counting like this? Because Because technically for the calculation of chromosomes, we are not counting chromatids, we are counting centromeres. This is a very important concept. Many students still, even though the syllabus is completed, still they are counting the chromatids and then they are saying ploidy. That is wrong. To say the ploidy, you need to count centromeres. Observe carefully. Now we will go with the concept. Now here, in the case of human, if 23 chromosomes are present, that is haploid. If 46 chromosomes are present, that is diploid. Yes or no? 23 chromosomes is haploid. If 46 chromosomes are present, that is diploid. And double to 46, that is 92 chromosomes if they are present, it is a tetraploid. One of this only you will find. You will not see any triploid here. You will see only haploid, diploid or tetraploid. That's it. Now come back to this. Now what is the question? Single thread like structures 23 are present. That means this is one chromosome. Like this, how many chromosomes? 23 chromosomes are present. That means what is the overall number of chromosomes? 23 only. If 23 chromosomes are present, what is the ploidy? Haploid. If condensed like this 23 are there, whatever it is one chromosome. Like this 23 are there, that means it is haploid. Clear? That means either like this 23 or like this 23. So what is the ploidy? It is haploid. Next one. Here, even though two chromatids are present, still it is considered as single chromosome only. So single chromosome like this 23. So if this is 1, like this 23 means total number will be 23 chromosomes. That means this is also haploid. This is also haploid, don't make mistake. And if they are present like this, here also, how many centromeres? Single centromere. Single centromere means this is one chromosome. Like this 23 means 23 centromeres. 23 centromeres means 23 chromosomes. It is haploid. Clear? Next one, come back to this. Here it is one centromere. Like this 46 means how many centromeres? 46 centromeres. That means what is the ploidy? So 46 chromosomes. So it is 2n diploid. Yes or no? Next one here also. So here it is also single. And like this 46, that means it is diploid. Next one here, double. It is a dyad. Dyad chromosome, but 46 are there. So it is one chromosome into 46. That means 46 chromosomes. So it is diploid. It is diploid. Like this you are observing. Understood or not? Suppose like this. So there is a condition like this. So this condition you are observing, actually you are observing for like this. Like this, if 92 are present. Like this, if 92 are present, then what is the ploidy? The ploidy will be 4n. Actually, this you are observing in the Interphase 2, in the Interphase 2, and even in the case of Telophase 2 also, I will tell you, okay, at the end of this one. So like this, if 92 are there, and or else for the same thing, you can write like single thread like 92, anything. If 92 are there, it will be 4n, that's it. And here dyad 92, nowhere you are observing, so I am not drawing that particular diagram. And this is how you need to write the ploidy. Very good. Next one. Next one we will talk about the C-value. I hope now you understood the ploidy. For the ploidy, you need number of chromosomes. How you are getting number of chromosomes? It is based on number of centromeres, not based on number of chromatids. Clear? Over. Now we will go with the C-value. C-value, it is the amount of DNA. Amount of DNA means how much of DNA is there. And for that, first we will see what is C, what is 2C, what is 4C. First you need to understand this. Now let us see what is the standard definition of C. Amount of DNA that is present in haploid content in the gametes. In the gametes, gametes are haploids. In the gametes, so with respect to human, each gamete will have 23 chromosomes. And in the gamete, what is the type of chromosome? It will be like this. So actually in the gamete, it will be like this or like this. Anything you can take, no problem. So like this, 23 are there. Now you just see it. Now to avoid the confusion, now I am removing this. Now let us see. Observe carefully here. Now this is one chromosome. Like this, how many chromosomes? 23 chromosomes are present. And in each chromosome, one double-stranded DNA is there. This is one double-stranded DNA. Yes or no? This is one DNA molecule. One DNA molecule. Like this, how many chromosomes? 23 chromosomes. So that's a total how many DNA molecules? So 23 DNA molecules. 23 DNA molecules. So in the case of human, if 23 DNA molecules are present, that is 1C. Then what is 2C? Amount of DNA should be doubled. Doubled means if 46 DNA molecules are present, if 46 double stranded DNA molecules are present, then it is called as 2C. Then what is 4C? If 92 DNA molecules are present, that is the 4C. Please understand here, it is the amount or content of the DNA. Understood or not? So in the case of human, this 23 molecules, now approximately it is 3 picograms. For the 2C it is double to this, right? So 3 double to the 3 means it will be 6 picograms. It will be 12 picograms. So in this way also amount, it is the amount of DNA. But at the level of this 11th and 12th, you don't need this particular value. But simply remember how exactly we are calculating. We are calculating the C value based on the number of DNA molecules. This is very important. If 23 DNA molecules, C, if 46 are there, it is 2C. If 92 are there, it is 4C. And now what you need to do? You need to link up this information with this condition. Now observe carefully. Now this is the condition, right? Either you just take this one or this one. I am talking for only one. Now in one, how many DNA molecule? It is 1 DNA. Let me change the color. So here it is having 1 DNA molecule. One is having one double stranded DNA molecule. Like this, total how many they are saying? 23 they are saying. So total how many DNA molecules? 23 DNA molecules are present here. Okay, next one. Here total 23 are present, but in this case each chromosome is having how many DNA molecules? Observe carefully. In one chromatid, this is double stranded DNA molecule. In the other chromatid, it is double stranded DNA molecule. That means in this dyad chromosome, how many DNA molecules are present? 2 DNA molecules are present. 2 DNA molecules are present. Each chromosome has 2 DNA molecules. Like this, how many are there? 23 are present. That means total how many chromosomes, how many DNAs are present? Total 46 DNA molecules are present. Observe this carefully. Here only one DNA molecule is present into how many chromosomes are present? 46 chromosomes are present. That means total how many DNA molecules? 46 DNA molecules are present. Yes or no? First understand the concept. Then you can easily write the C value. Next one here. Here 2 DNA molecules are present in each chromosome. Like this, how many chromosomes are present? 46 chromosomes are present. So 46 into 2 it is 92 DNA molecules are present. Yes sir. And the last one here, one DNA molecule is present. Like this 92 are present. That means total how many DNA molecules? 92 DNA molecules are present. Now based on the DNA molecules, tell me. So just now let us quickly revise. If 23 DNA molecules, C. 46 DNA molecules, 2C. If 92 DNA molecules, it is 4C. Now come back to this table. Now tell me. 23 DNA molecules. So it is C, means 1C. Okay, it is 1C. Now here 46 DNA molecules. So it is 2C. Next one, here also 46 DNA molecules. So it is 2C. Next one, here it is 92 DNA molecules. So it is 4C. And here also it is 92. So here also it is 4C. Understood or not? Please observe carefully. Now what is the overall conclusion here? There is no relation between Ploidy and C value. Don't link these two. Some students, what they are thinking, if it is haploid, it should be C only. If it is diploid, it should be 2C only. How you will say this? Even though it is haploid, here you are observing 2C. Understood or not? And here, even though it is diploid, here you are observing 4C. So that's why don't link these two concepts. Ploidy is an individual concept, C value is a different individual concept. Don't link these two. Don't link these two. And one more thing, when they are saying N by C value, don't write this N and this C. That N by C is nucleocytoplasmic volume ratios. That is a different concept. All these are individual concepts. Don't interlink these things. Study these things individually. That's it. Okay. So where to interlink, where to keep individually, that also you need to know. This is what the basic concept. And now, if you know the diagram, so what type of chromosome, whether it is present as a chromatin thread-like structure, or condensed chromosome. That you need to know. For each and every phase, you need to know whether it is like a thread-like structure or condensed rod-like structure. That you need to know. And how many are present. This number also you need to know. And once you know this information, you can easily write the Ploidy and C-value. You will never ever make any mistake. So this is the most important concept. And even those students, generally those who are scoring more than 340 out of 360 for biology, so even those students are also making mistake in these concepts. So this is a very basic, general, important concept, so that you should never ever make any mistake in these particular basic things. By simply looking at the diagram, you need to clearly and correctly say what is the Ploidy and what is the C-value. So you need to practice in such a way. Anyhow, for your understanding purpose, at the end of the lecture, once again I will make the complete table with all the diagrams, though it will take a little bit more time. So please be patient. We are doing all these things for you only. And this is the reason why we are keeping it public also. Okay, so anyhow, those who are taking the app access by payment, so anyhow you will get these videos, but some students still they are making mistakes. So that's why we thought that, so at least in this revision lecture, at least some basic information, 70 to 80% basic information is general and every student can understand and remember that. But the remaining 20 to 30% is the most important and crucial concept, and where generally students are making mistake, and those mistakes are responsible for not getting the better ranks. So that's why at least in the revision lectures we are focusing on those 20 to 30%, and remaining 70-80% quickly we can revise also, and there we don't need to focus more. Clear? Okay, now this is what the general basic information. I hope now you understood what is Ploidy and what is C-value, and you should not make any mistakes. At the end of the lecture, quickly and simply by looking at the diagram, I will write all this, and there you should not say that and how this fellow has written this Ploidy value and all the things. This is the basic information. Clear? And if you are having any doubt, stop the video here and go back to that Ploidy and C-value concept once again and get better clarity, and then continue the video again. Clear? Now let us continue further. And now we are going with the mitosis, and here I don't want to draw any diagrams here. Quickly I will explain the concept. Anyhow, at the end of the lecture I will draw each and every phase with the diagram and concept. Clear? Now under the mitosis, first we are observing what are the various phases that are present in this. In the case of mitosis, first we are talking about the interphase and then cell division phase. This is the cell division proper, and this is the interphase. Within the interphase again, three subphases are present. What are those? G1 phase, S phase, G2 phase. These are standard and fixed sequences. G1, S, G2. These three are part of interphase, and then M phase will come. Within the M phase again, two phases are present. One is called karyokinesis and the next one is called as cytokinesis. Within the karyokinesis again, first one is prophase, next metaphase, next anaphase, and then telophase. So what is the overall sequence here? G1, S, G2, prophase, metaphase, anaphase, telophase, cytokinesis. And once again two cells are produced in mitosis, and again they will follow the same cycle. That's why cycle concept is used only for mitosis. And since there is a fixed sequence, already I told you, I made a separate video on how to understand NCERT lines, how to observe this. So there in that video I told you when to use or how to use follows, the word follows and followed by. Suppose I am asking a question that S phase follows, which one you will write? S phase is followed by. Likewise, question can be asked for any of these. So how to use those? Follows, followed by, precedes, preceded by. These four words are very important. I told all these things very clearly in that separate video. So please go through that basic introductory videos. They are available even on the YouTube also. Anyhow, they are there in the app. They are available on YouTube also. So please go through that first. Many students, you know everything. But if you don't know what is the meaning of follows and followed by, precedes and preceded by, definitely you will make a mistake. So please observe this. Next one is regarding the cell cycle. So here first we are talking about the interphase. So interphase is approximately having the time of 95% duration of the overall cell cycle phase, and then it is the phase between two successive M phases. What is the meaning of inter? Inter means between. Between what? Between two M phases. So here you are saying, now this is also one concept. Here you are saying that G1 phase, S phase, G2 phase, and then M phase. But only single M phase is there. If single M phase is there, how you will say that interphase is the phase between two M phases? How you will say that two M phases? Now observe this concept carefully. After the M phase, see, initially we are starting with single cell, and this single cell will divide and form two cells. Yes or no? And for the newly formed cells, again they are going with, this is the G1 phase, they are going to S phase, G2 phase, M phase, and then finally it is forming. If you are talking for one cell, again it is forming two cells. First cell there is an M phase. For the second cell also there is an M phase. So between M phase of first cycle and M phase of second cycle, there is a stage, and this stage is called as interphase. So M phase of first cycle, M phase of second cycle. In between this there is a phase, and that's why it is called as interphase, between phases, between two M phases. So between two M phases of two successive cell cycles. So this is what the meaning is. Try to understand the meaning. So that's why they are saying that it is the phase between two successive M phases, two back-to-back M phases. Okay, this is what they are saying, and it is called a resting phase. Why it is called as resting phase? So it is called as resting because in the interphase, cell is not dividing. Cell is not dividing, that's why it is resting phase. It does not mean that it is metabolically inactive. Many students are thinking that resting means metabolically inactive. What is the significance of word called metabolically active, metabolically inactive? Simple logic. If a cell in a phase G1, S, G2, M phase or G0 phase, whatever it is, observe carefully. When a cell is living, it should be metabolically active, or reverse is the better sentence. If a cell is metabolically active, then only it will be living. If a cell is metabolically inactive, means it will die. So that's why whether it is G1 phase, S phase, G2 phase, M phase, or even G0 phase, though it is not dividing, in all these phases the cell is metabolically active. Metabolically active, don't make any mistake. So cell is metabolically active in interphase, but why it is called resting phase? Because it is not dividing. That's why even in G0 phase also we are saying inactive, because it is not dividing. Please understand these sentences. This is what the relation is. Next one. Then if it is not dividing and if it is occupying 95% of the time, then what exactly it is doing? It is getting prepared for the actual cell division in the M phase. It is getting prepared, and during this phase what happens? So here cell growth is taking place, and even the DNA replication is also taking place. And it is divided into three subphases: G1, S and G2 phases. G1 is the gap one, S is the synthesis phase, G2 is the second gap phase. Okay, next one. Regarding the M phase, it is also called as mitosis phase, and this is the cell division proper. Why it is called cell division proper? Because actual cell division occurs in this particular phase. That's why it is called cell division proper. So here 95% time is interphase, means around 5% is M phase. And here in NCERT they are saying that with respect to human cell, overall cell division generation time is 24 hours, but actually M phase is taking only 1 hour. Only 1 hour it is taking. That means interphase is taking approximately 23 hours. Based on the sentences, you need to interpret opposite sentences. Okay, next one. And this is where actually the cell division occurs, and this is what the mitotic phase is. And again it is having karyokinesis, cytokinesis. In the karyokinesis what happens? Nuclear division occurs. In the cytokinesis, cytoplasmic division or distribution into daughter cells will occur. And within the karyokinesis again 4 phases: prophase, metaphase, anaphase, telophase. So these are the various phases that we are studying under mitosis. Clear? First one is the gap one, first gap phase. So what is the location of this? What is the location of this first gap phase? It is between M phase and S phase. Okay, some students they are writing reverse, S phase and M phase. You should not write like this. Between S phase and M phase means what will come? G2 phase will come, because this cycle always you need to see in this clockwise only. Between M and S means G1 will come. Between S and M means G2 will come. So don't make mistakes here. First gap is between mitotic phase, means actual cell division, and S phase means DNA synthesis phase. Between this G1 phase is there. If the sentence is reverse, what will be the answer? Second gap phase is the answer. So you need to observe these things also, minute things also you need to observe. Next one. So here also cell is in all the stages cell is metabolically active, and here cell growth, it is a continuous process. Cytoplasmic increase is a continuous process that is taking place, and here DNA replication does not occur. And with this regular regular sentences information is completed. But in the case of NCERT, under the heading of summary at the end of the chapter, they have given some extra sentences. What they are saying about the G1 phase? Most of the organellar duplication occurs in the G1 phase. Very, very important thing. You need to compulsorily study summary for this chapter. Many sentences they gave extra in the summary. Okay, most of the organellar duplication occurs in the G1 phase. Majority of the organelles are duplicating in G1 phase. And here itself I will tell you, in the S phase, you are talking about centriole or centrosome duplication. S phase, centrosome duplication. Chloroplast and mitochondria duplication you are studying under G2 phase. Observe this. Chloroplast mitochondria duplication you are studying under G2 phase, centrosome S phase, and majority of the organelles are duplicating in the G1 phase. Don't make mistakes, observe carefully. Next one. S phase, it is also called a synthetic phase. What is synthesized here? DNA is synthesized in this particular phase. So please observe this. DNA is synthesized. So sister chromatids are formed. So please remember, only DNA cannot form the chromatid. Along with the DNA, histone proteins are also required. So within the S phase, DNA replication or DNA duplication, duplication, both the meanings are same only. Some are giving different meanings for this. That is wrong. Duplication, duplicate means what? Another copy. Replica means what? Another copy. So replication, duplication, both the words you can use for DNA and both the words you can use for chromosome. Understood or not? So DNA replication or DNA duplication occurs in S phase. Not only DNA replication, histone protein synthesis also occurs, because we are talking about eukaryotes, right? So in the eukaryotes, along with the DNA, histone proteins are associated. Then only association of histones with DNA only forms chromosomes. So in this way, you are talking about the sister chromatids. Why you are saying sister chromatids? Please remember, in the G1 phase, it is a thread-like structure. And within this, double stranded DNA is present. Now in the S phase, what happens? Double stranded DNA, both the strands, already in molecular basis of inheritance we studied, both the strands will separate and a new strand will be synthesized. So this will become one double stranded DNA, this will become one double stranded DNA. One double stranded DNA is present in this, other double stranded DNA is present in the other copy. So now here we are, what you are saying? So DNA is present, histones are also present. DNA and histones means what? Chromosomes. So in the general sentence, if you say chromosome, this is chromosome and its copy. That's why you are saying chromosomal duplication, chromosomal replication. Any word you can use, but generally we are using chromosomal duplication occurs in which phase? S phase only. DNA replication occurred means chromosomal duplication also occurred. So this sentence was clearly mentioned in the summary. So observe carefully. Nice. So even though it is a copy, in the regular sentence they said that DNA replication occurs. DNA content, amount of DNA will double because another DNA was formed. So C value doubles. In the G1 phase it is 2C. In the S phase it will become 4C. So DNA amount doubled. But the ploidy remains same, number of chromosomes remains same, because both the sister chromatids are attached at the same centromere. Initially also single chromosome, now also single chromosome, because centromere remains same. Please remember, for ploidy calculation, I told you just now, for ploidy calculation, centromere is important. So centromere is same only. So that means it is considered a single chromosome only. Though it is single chromosome, it has a two chromatids. These two are exactly same, 100% exactly same. That's why these two are called as sister chromatids. Sister chromatids. So don't make mistakes, observe the concept very clearly. So this is what the basic information is. Clear? This is what they are saying. So here, ploidy remains same, number of chromosomes remains same, but the amount of DNA or the DNA content doubles from 2C to 4C. Clear? This is what they are saying. In the G1 phase, 2N ploidy is there. So in S phase also 2N ploidy. Ploidy remains same, amount of DNA is getting doubled. This is what. Specifically for animal cell, what we are saying, centrosomes. Within each centrosome, two centrioles are present. So they will undergo division. So that's why, so in the G1 phase, only one centrosome with two centrioles are present. But in the S phase, two centrosomes means four centrioles are present. So centrosome duplication is also occurring in the S phase. Next one is regarding the second gap phase. What is the location of the second gap phase? Between S phase and M phase. You need to write like this only. Okay, this is what. And here exactly what happens. Cell is getting prepared for next M phase. Please remember, in the M phase what happens. So here they said that proteins required for M phase proteins required for M phase are synthesized in G2 phase. Why it is like this? What is the logic here? Because in the M phase, chromosomes will undergo condensation, means DNA is undergoing condensation. If DNA undergoes condensation, it will be tightly packed. Tightly packed is heterochromatin. I am linking up with molecular basis. Tightly packed is heterochromatin. Heterochromatin is transcriptionally inactive. Transcription does not occur. RNA synthesis does not occur. So in the M phase, because of chromosome condensation, transcription does not occur. mRNA is not produced. If mRNA is not produced, there is no protein synthesis. That means within M phase, M proteins cannot be produced because it is in the condensed form. So that's why proteins required in the M phase should be produced in advance. In advance means which phase? G2 phase. This is the logic, interlink it. So now what is the standard example? What are the important proteins required for the M phase? For spindle fiber formation, spindle fibers are nothing but microtubules. Microtubules are nothing, means what? Tubulin protein is the important protein. So that's why the majorly in the case of G2 phase, tubulin protein is synthesized. And this tubulin protein will be used in M phase for spindle fiber formation, so that spindle poles, spindle apparatus will be produced. Yes or no? This is what the interlinked information. Next one, cell growth continues. Cell growth is taking place in G1 phase, S phase, G2 phase, all the three phases. But read the summary. In the summary, they said that G2 phase is the period of cytoplasmic growth. What is the meaning of this? Cytoplasmic growth is occurring in G1S and G2 phases. Still, they said that it is the period of cytoplasmic growth. So, what is the conclusion based on this particular sentence? The meaning is cytoplasmic growth or cell growth is occurring in all the three phases, G1S and G2, but maximum growth is occurring in the G2 phase. Experimentally, scientists have proved that the major volume increase for cytoplasm is occurring in G2 phase. This is what the meaning of this particular sentence. So please observe each and every sentence. And next one is, in the NCRT, in a box, they have given some questions, but they have not given answers. Let us quickly go through this. How do plants and animals continue to grow in all their lives? How they are able to grow? Because of cell division. Cell division is taking place, that's why they are able to grow continuously. Next one, do all cells in all plants divide all the time? So do all the cells in the plant divide all the time? No, all the cells do not divide, because you know that some cells after some divisions and then they are start growing and then they are becoming matured. So once they are becoming matured, generally they will not divide. Yes or no? So in this way they are not continuously dividing. So answer will be no. Clear? And here, do you think all the cells continue to divide in all the plants and animals? No. No. Here also no. But in the case of majority, not all animals, in the majority animals, once a particular growth is achieved, then growth further it will not occur. Further growth does not occur. Okay. But in the plant cells, yes, plants are able to divide continuously in their lifetime, but all the cells in the plants do not divide continuously. Generally, this sentence is applicable to meristematic cells. Meristematic cells present in the plant cells, yes, they are able to divide continuously in their lifetime. But the remaining cells, they are not continuously dividing. But if as a whole complete plant, if you are saying, yes, plants, they are able to grow. As a complete organism, you are saying, so they are able to grow in the lifetime. This is what they are saying. Okay. Next one, can you tell the name and the location of tissues having cells that divide all their life in higher plants? In the higher plants, there are some cells that are present in the meristems, meristematic cells. They are able to divide continuously in their lifetime. Next one, do animals have similar meristematic tissues? Exactly similar meristematic tissues are not there, but in the case of animals, there are some cells called as stem cells. And these stems are also comparable to, stem cells of animal cells are comparable to meristematic cells of the plants, and these stem cells are helping in the growth of the animals. Okay, understand this. This is what. And then in a separate box in the NCRT, they have given this information. Here directly I am giving the answer, but how exactly you are getting this answer? So already in the beginning of this particular lecture, I told you how to calculate Ploidy and the C-value, right? If you know that, you can directly give these answers. If you don't know, anyhow at the end of this chapter, I am giving the each phase diagram, right? Based on that, you can easily write this. Clear? Now let us see what they gave in the onion root tip cells. So actually it is a diploid, and here it is having, in the diploid condition, it is having 16 chromosomes. So in the haploid, how many will be there? 8 chromosomes will be present. Now in that case, now in the G1 phase, how many chromosomes? 16 chromosomes will be there. That means diploid. In the S phase also it is diploid. G2 phase also it is diploid. After M phase means what? After M phase again, G1 phase only will come. So after M phase means again G1 phase only. This is what the phase. So this is what the ploidy, this is what the number of chromosomes. Clear. Next one is regarding DNA content. In the G1 phase 2C, S phase it is 4C, though it is 2N, C value is 4C. And here G2 phase also it is 4C. After M phase means once again it is G1 phase, so again it is becoming 2C. So this is what some basic information. So read each and every question of NCERT, and not only for this, once they have started asking question like this, they can change any organism, they can give the name of any organism here, they can give some basic information that so and so number of chromosomes are there, and now they can link up this with any stage of the mitosis, any stage of the meiosis, anything they can ask. You should be careful. So that's why you should know the diagram, phase diagram. Once you know the diagram, you can easily write Ploidy and C-value. And once you know what is the basic Ploidy value, and you can substitute that value, that's it. And you can get the answer for any question. So in this way, they are giving only some basic information, and it has some huge application level. So that's why you need to understand the basic concept. Clear? So in this way, we are talking about the division of the cells, but still there are some cells, there are some mature cells, they are not generally dividing, and the major examples are heart cells, RBCs and neurons. These cells are not dividing. Next one. So generally, why this particular cell division is taking place? Cell division, cell cycle, cell division means mitosis. With respect to mitosis we are talking. Generally this mitosis is taking place to replace the cells that are injured or that are dead due to various reasons. Clear? This is what the major reason is. Next one is regarding the G0 phase. It is called as a quiescent phase. So what exactly happening in this G0 or quiescent phase? It is a quiescent, it is inactive phase. It is inactive phase. So generally cells in this particular stage, they are coming out of the G1 phase. Once the M phase is completed, now the cells will exit the cell cycle. They are coming out of the cell cycle and they are temporarily they are present in G0 phase. So in the G0 phase, what is happening? They are not dividing, not dividing, not dividing. That's why it is inactive. Not dividing, that's why it is inactive stage. But since it is a living cell, it is metabolically active. Don't make mistake. It is metabolically active. That's why it is able to live. And then generally it is not dividing. And here they gave a sentence. So they no longer proliferate. That means they do not divide unless called on to do so depending on the requirement of organism. Depending on the requirement of organism. If they are not called, please understand the meaning. If they are not called for division, they will not divide. Because they are not called for division. That's why they are not dividing. If they are called for the division, then they will divide. Please understand the meaning of this. So why they are not dividing? Because there is no requirement. Since there is no requirement, they are not dividing. Suppose this is the let us talk about this outermost layer of the skin. Now here it is normal. So now there is no requirement for the division. So now the cell in this particular one will be in the G0 phase. Suppose I have fallen down, and then here there is some damage of the cells. Cells got damaged. Now these cells, damaged cells will be removed. And once the damaged cells are removed, now there is a requirement to replace this. So if there is a requirement means what? Now these cells will be called for the division. So now what happens from the G0, again they will come into the G1. At the end of the G1 they will enter, and then they will go into yes G2M, and then now they are forming the new cells. So in this way this cycle will be continued. Until when it will be continued? Once it becomes normal. Until then they are keep on dividing and dividing so that they are replacing all the damaged cells. And once it got replaced, now there is no requirement. If there is no requirement, once again they will go into G0 phase. This is what the meaning is. If there is no requirement for division, cell will be temporarily present in the G0 phase, inactive phase. That is called quiescent phase. If there is a requirement, then they will be called for this division. Then it will again enter into the cell cycle, and it will be keep on dividing and dividing until that particular requirement is finished. Once the requirement is finished, once again it will go back to that quiescent phase. This is what the concept of that quiescent phase. Clear? Next one. Where does this mitosis occur? Since we are talking about the M phase, mitotic phase, directly I am talking about all the information here. So where actually this mitosis occurs? So mitosis generally occurs in the diploid somatic cells. But please remember, mitotic division is called as somatic division, right? Even though mitosis is called as somatic cell division, it is still occurring in reproductive cells. Anyhow, in this lecture only, at the end I will show you about the human gametogenesis. So anyhow, the diagram I will show you. If once you see that one, anyhow you studied about the human reproduction chapter. Spermatogonia, oogonia concepts we studied. Initially, this spermatogonia, oogonia will divide by mitosis only. So spermatogonia, oogonia are the reproductive cells, germ cells, yes or no? Please remember, gamete mother cells are also called as germ cells. Gametes are called as germ cells. They are the haploid germ cells. And gamete mother cells, the cells from which these gametes are produced, they are also called as the germ cells. But they are diploid germ cells. Understood or not? These concepts clearly I have explained in the cell cycle, cell division chapter in the regular classes. So if you still have any doubts, you please go through that. Now let us come back to this one. So even though mitosis is called as somatic cell division, it is occurring in somatic cells and even in the reproductive cells. Understood or not? So now, if it is occurring in both the types of cells, why mitosis is called somatic cell division? Now you need to think about this. Why mitosis is called as somatic cell division? Because somatic cells divide by mitosis. Somatic cells divide by mitosis. But reproductive cells, they are having both mitosis and meiosis. Reproductive cells, they are undergoing both mitosis, meiosis. But somatic cells are dividing only by mitosis. So that is the relation between this mitosis, why mitosis is called as somatic cell division. Understand this. Next one, in the case of male honeybees, generally gametes are produced by meiosis process. But in the case of male honeybees are haploid cells. If they are already haploid cells, it cannot undergo meiosis. So in that case, it is undergoing mitosis to form this particular gametes, male gametes called the sperm cells. So in the case of male honeybees, male gametes called sperm cells are produced by mitosis, not by meiosis. Very important sentence. This was mentioned in Principles of Inheritance and Variation chapter also. Next one. So generally in the case of animal cells, we are observing this mitosis in the somatic cells, generally. But in the case of plant cells, we are frequently observing mitosis in both haploid and the diploid cells. Clear? So that's why in the case of, mainly in the case of plant cells, the haploid pollen grains, now they are undergoing the mitosis to form the male gametes there. So that's why frequently we are observing this particular concept here. And next one, gametophyte of bryophytes, and their haploid, now it is producing gametes by mitosis only. Okay, these are some important examples where generally we are observing the mitosis. And now we are talking about, till now we studied about the interphase G1S and G2, and now we are going with the M phase. Now M phase is called mitotic phase. And why it is called as equational division? So mitotic phase or the M phase, or in general it is about the mitosis. Mitotic division is called equational division because the ploidy of parent cell and the ploidy of daughter cells are exactly same. If the parent cell is 2n, daughter cell is also 2n. If the parent cell is n, daughter cell is also n. So since the ploidy of parent and daughter cells are exactly same, it is called as equational division. So that point also you need to remember. Next one. So it is a progressive process. Don't think that after every stage it is having some gaps or pauses. It is not like this. It is a progressive, continuous process. For our understanding purpose, we are thinking that okay, from here to here it is one phase, from here to here one phase. For our study purpose, we have differentiated, but it is a continuous process. So that's why clear-cut lines cannot be drawn. So that's why sometimes what happens is one point can be discussed in one particular phase, but in some other reference books, the same point may be discussed in some other phase because clear-cut lines cannot be drawn here. So according to our understanding purpose, for easy study only, we have separated them like this. That's it. Next one is regarding the karyokinesis. Within the M phase, first one is the karyokinesis. In that first we will go with the prophase. And regarding this prophase, the spelling is very important because so in the cell cycle phases we are using prophase, P-R-O-P-H-A-S-E, S-E. But here there is another word that is called prophase, P-R-O-P-H-A-G-E, prophase. Please remember, phages are the viruses. Phages are the viruses. So this prophages, this is the viral DNA that is present within the bacterium. Viral DNA that is associated with bacterial DNA is called as a prophase. PHAGA, prophase. That spelling and meaning is completely different. And here under the cell cycle we are using the spellings called as phases. PHASE phase. It is not PHAGE phase. Please understand this. So it is the prophase. It is the first phase of karyokinesis we are observing in the mitosis. And this is happening after G2 phase. Followed by words. You should clearly understand this. Next one. And what exactly happening in this prophase? In the prophase, what happens is condensation of this chromatin, because in the interphase extended thread like chromatin is present, but in the M phase it is undergoing condensation to become rod like structures. So now that condensation is beginning in this prophase, and the nuclear membrane will undergo start breakdown. So at the end of this prophase, that nuclear membrane will be completely disappeared. Nuclear membrane will be completely disappeared. So endoplasmic reticulum, Golgi complex, nucleolus will be disappeared. Now you need to understand, okay, for this condensation is occurring, okay, but why nuclear membrane, endoplasmic reticulum, GC, Golgi complex are undergoing disappearance? They are also undergoing breakdown and they are disappeared from that location. But actually these fragments will go just below the plasma membrane. Why they are going there? The logic is, in this particular M phase, the chromosome will undergo condensation and they are moving to opposite poles. If they have to move to opposite poles, the area should be clear. If nuclear membrane is there, how the chromosomes can move to opposite poles? So that's why nuclear membrane should be broken down. And Golgi complex and endoplasmic are large network-like structures in cytoplasm. If large network-like structures are there, how chromosomes can move? So that's why ER, GC and nuclear membrane should undergo breakdown and they should move away from that position. So that's why these fragments are going just below the plasma membrane. Understand this. So since anyhow nucleus is not there, nucleolus also will disappear. These are the logical points. And next one. And next important point that we need to understand regarding the prophase is, in the case of prophase, what happens is centrosomes are moving to opposite sides. Centrosome duplication occurred in S phase and now they are moving to opposite poles. And now what happens is microtubules will be started forming from this. If centrosomes are present, asters will be observed. Understood or not? And along with this, the longer microtubules called spindle fibers are also present. So now you need to understand is, in the case of animal cell, centrosomes are present and then asters are present. But in the case of plant cell, centrosomes are not present, so asters are not present. But in both plant and animal cells, spindle fibers are present. Please understand these points. So that's why spindle fibers, spindle poles, these terms are used in both plant and animal cells. But centrosomes, asters, these terms are used only for animal cell, not for the plant cell. So that's why anastral, without asters, the cell division is taking place in the case of plant cell. And eccentric, why it is called eccentric? Because centrosomes and centrioles are not present in plant cells. So that's why centric division in animal cell, eccentric division in the plant cell. Astral cell division in animal cell, anastral cell division in the plant cell. Understand these relations. Don't try to by heart. Understand the concept, then it will be easy for you. So this is what happening. Clear? And during this process, you need to also understand that spindle fibers are forming in the prophase itself. But please observe the NCERT diagram carefully. Already in the cell chapter, revision part 2, I told you under the centrosomes, spindle fibers are coming from the superficial surface of this particular centrosome. These spindle fibers are not directly attached to centrioles. Please understand the concept. This is what it is. Okay. In the case of plant cells, even though centrioles are not present, centrosome is not present, some proteins will gather together and they will help in microtubular organization. Okay, so this is what actually happening in the case of plant cells, but these details were not clearly mentioned in NCERT, but some basic information you need to understand for more clarity. Okay, this is what about the prophase. So prophase is complete. So at the end of the prophase, what happens? Nuclear membrane will break down, all these things are taking place. Nice. That's what the point I wanted to tell you. Next important point is, in the NCERT they used a word called mitotic apparatus. You need to understand this. In the cell cycle cell division chapter, they used a word called as mitotic apparatus. But in the cell chapter, under the heading of centrosome and centrioles, there they used a word called as a spindle apparatus. Spindle apparatus. So mitotic apparatus and spindle apparatus, both of them are same only. Then what is this spindle or mitotic apparatus? So this spindle or mitotic apparatus is nothing but both the spindle poles plus spindle fibers. These are the spindle fibers. Both the spindle poles plus spindle fibers, all these things are collectively called as one mitotic apparatus. So please remember, for the calculation purpose, they may ask this: one mitotic apparatus. What is one mitotic or one spindle apparatus is? One spindle apparatus is two spindle poles plus spindle fibers. So entirely it is called as spindle apparatus. That's why for the spindle apparatus, suppose this is one side, this is the other side. Completely it is called as spindle apparatus. Clear? This is what another important point that they are talking under the prophase itself. These are some important things that they are saying here. Next we are going into the metaphase. Now this condensation started in the prophase, so that now you can start seeing them under the microscope. So because of the condensation, what happens is, now initially in the interphase, all these are like thread-like interconnected tangled structures. Now in the prophase, now they are separated. So untangling will occur in the prophase, and the condensation started in prophase, and the condensation is keep on continuing till metaphase. And the chromosomes in the in the mitosis, with respect to mitosis, if you talk, maximum condensation we are observing in the metaphase. Suppose anyhow in the next part we will, not next part means after few minutes we will study about meiosis. In the meiosis 1, where exactly we are observing the most condensed? In the meiosis 1, fully condensed chromosomes are seen in diakinesis. Diakinesis, that is the last subphase of prophase 1. But in the mitosis, the maximum condensation of chromosome we are observing in the metaphase. So since they are maximum or most condensed, now here we can easily study morphology or structure of the chromosome. Don't write shape. Shape of chromosome, V shape, J shape, I shape and L shape, these shapes we can see in the anaphase. Sorry. So shapes of chromosomes are easily studied in anaphase, but structure or morphology of chromosome is easily studied in metaphase. Please understand these basic and important points. Not only this, and here they are saying that these chromosomes are formed directly in cytoplasm. Why you are saying like this? Chromatin in the interphase is present inside the nucleus, but when condensed chromosomes are formed, at that time nuclear membrane is not there. So these condensed chromosomes are formed directly in the cytoplasm. This is another technical important point that you need to understand. So these are some important points regarding the metaphase. And in the metaphase, what else is happening? So spindle fibers will attach to kinetochores of the chromosomes, and here what happens is all the chromosomes will be aligned side by side. All the chromosomes will be aligned side by side, and this particular location is called as metaphase plate. Metaphase plate or equatorial plate. So all these chromosomes will be aligned at the center of, I will give you two options. Both the options were mentioned in NCERT, but in the examination, if they are giving both the options, you need to select only one. So which one you will select? Now I will repeat the question once again. During the metaphase, all the chromosomes are aligned at the center of, first option, center of the cell. Second option, center of the spindle apparatus. In the NCERT, both the sentence they give. In general, if you see, both of them are correct. But if both the options are given, you need to select only one. And which one is the most correct one? Observe carefully. In the metaphase, all the chromosomes are aligned at the center of spindle apparatus. This is the spindle apparatus, right? Center of spindle apparatus. Generally, center of spindle apparatus is center of the cell. So that's why both of them are correct. But most correct is center of spindle apparatus. In some cases, here is one spindle pole, here is one spindle pole, and here is the center of spindle apparatus. But that is not the center of the cell. So that's why center of spindle apparatus is more appropriate than center of the cell. So please observe this particular important point. And here Diad chromosomes are there, spindle fibers will attach to kinetochores, and now these spindle fibers will bring all the chromosomes to center of the spindle apparatus. Clear? Next one. Next one is regarding the anaphase. During the anaphase, what happens? Splitting of centromere will occur. Here centromeres are present here. Splitting of centromere will occur. So please remember, once splitting of centromere occurs, now each sister chromatid will get its own centromere. Initially here it is called as one chromosome. This is called one chromosome because one centromere. But the same thing now it is called as two chromosomes because each sister chromatid is having its own centromere. So here one centromere, here one centromere. So on this side how many will go? With respect to human if you are talking, 46 are present like this. But in the anaphase what happens? 46 single chromatids are going on one side, 46 single chromatids are going on other side. And the important point is 46 centromeres on upper side, 46 centromeres on bottom side with respect to diagram I am saying. Okay. So 46 plus 46 will be 92. So what is happening to ploidy? Ploidy is getting doubled in the anaphase. Please understand this. Anyhow, separately under the diagrams you will get more clarity. And here what happens? Splitting of centromere will occur. Sister chromatids, now they are called as daughter chromosomes. They are moving to opposite poles. When they are going to opposite poles, centromere is facing the pole. So that arms are coming behind, arms are trailing behind. And here in the anaphase, you can easily study the shape of chromosomes. For metacentric, V shape, submetacentric, L shape, acrocentric, J shape, telocentric, I shape, you can easily see in the anaphase. This is what the important point that they are saying. Okay. Next one is regarding the telophase. In the telophase, what happens? In the anaphase, still DNA is condensed, still chromosomes are condensed, but in the telophase, what happens? Now they will undergo decondensation. Once again, they are becoming thread-like structures. So with respect to human, 46 one side, 46 other side. And around this group, again nuclear membrane will be formed. Endoplasmic reticulum Golgi complex will be formed. All these things will be again formed. And this is what exactly happening in the telophase. In simple, you can say opposite to prophase is taking place here. And here in the telophase, since nuclear membranes are formed, in a single cell, two nuclei are present here. Two nuclei are present here. And this is what happening in the case of telophase. In simple, I am saying. And next one is regarding the cytokines. Now cell division, cytoplasmic division should occur so that now the cell organelles and nuclei should be distributed to daughter cells. And now here in the case of animal cells, what happens? It is happening by cell furrow method or cleavage method. Cell furrow method or cleavage method is taking place here. And this cell furrow or cleavage is taking place starting on the surface. It is starting on the surface and finally it is reaching to the center of the cell. Since it is starting on the surface or the periphery and going to the center of the cell, this type of movement is called as centripetal growth. Centripetal growth. So this furrow is extending by centripetal growth, so that finally once it is meeting at the center, now the cell will be dividing into two daughter cells. This is how cytokinesis occurs in the case of animal cell. But in the case of plant cell, what happens is some microtubules. I am not going into more details because it is complicated mechanism. Some microtubules and some vesicles, some components, now they are arranged at the center of the cell. Initially here what happens? Opposite. In the animal cell cytokinesis, furrow is starting at the surface and coming to the center. But here cell plate will be formed at the center. Now it is extending to the outer side till it is touching the existing cell walls. In this way it is keep on growing and finally dividing the cell into two. So because of the cell division, what happens is now the cell organelles are also distributed between the two daughter cells, and the two nuclei also will be distributed between the two daughter cells. In this way finally at the end of cytokinesis, two daughter cells are produced. But the important point is the ploidy of each daughter cell is exactly same like ploidy of parent cell. That's why it is called as equational division. Clear? Next one. The cell plate that is formed during the plant cell cytokinesis, this will slowly matures into middle lamella. That's why middle lamella is acting as is helping in holding or gluing the adjacent cells together. This point already in the cell chapter we have studied. Next one is regarding the multinucleate condition. Suppose, see, first karyokinesis will occur so that nuclei number will be doubled and then cytokinesis will occur. Suppose cytokinesis does not occur. Suppose cytokinesis does not occur, then what happens? In a single cell, many nuclei will be present and this will cause multinucleated condition. And now here in the NCERT they are talking about the syncytium. So now here they have given an example, that is liquid endosperm in the case of coconut. This is the standard example, so try to remember this example. Next one is regarding the significance of mitosis. Already we studied. So it is occurring to maintain the nucleocytoplasmic ratio. And second one, generally it is occurring in the diploid cells, but it can also occur in the haploid cells. Next one, in the case of lower plants and in the case of some social insects like honeybees, even in the case of males also, haploids also, mitotic division is happening to produce the gametes. This is also they are saying. With respect to honeybees, if you are talking, queen is diploid fertile female and here meiosis occurs to form the ova. But in the case of males, they are the haploid and fertile males. They are also called as drones, and here sperm cells are produced by mitosis, not by meiosis. Okay, and worker bees are females, but they are sterile females, so they cannot produce the gametes. Clear? Next one. And here the ploidy of daughter cell and parent cell will be remaining same, and mitosis is occurring in multicellular organisms so that growth will occur, and even it is used for the repair of the cells. And here in the case of mainly for the plants and many things, now it is helping in vegetative propagation also. Clear? Next one. And in some cases cells are continuously in this mitotic division so that they are replacing the damaged cells. So generally the cells of the upper layer of the epidermis, so now they are continuously in the division form, and then lining of the gut. So in our gut also the lining is continuously damaged because of the food and some of the chemicals that are secreted. So that's why these cells are also continuously in division for the replacement, and then blood cells are being constantly replaced. And in the case of meristematic tissues of the plants also, the divisions are continuously taking place so that now they can help in the continuous growth of the organism. So this is some basic information. I don't need to spend much time on this. And with this, mitosis concept is completed. Now we are going into the meiosis concept. And here this is generally occurring in the sexually reproducing organisms, generally for the formation of the gametes. Okay, so this is what generally happening here. So that now please observe carefully. When meiosis occurs, now what happens? The ploidy, whatever the ploidy that is present in the parent cell, so that will be reduced to half in the daughter cells. And please remember, meiosis does not occur in haploid cells. So it should occur in the diploid cells only. Mitosis is occurring in both haploid and diploid cells, but meiosis occurs in diploid cells. So when the parent cell is diploid, so the daughter cells will be off of this. Off of this means what? Haploid cells only. This is what point. Anyhow, separately in the NCERT they gave 4 important points. There also they said. There also once again I will tell you. Next one. Diploid cells form haploid gametes by meiosis. Next one. In the case of meiosis, what happens? Haploid cells are produced. That's why it is resulting in the formation of haploid phase in the life cycle of that particular organism. But when it will be restored into diploid phase, when haploid gametes are undergoing fertilization, once again diploid phase will be restored. Okay. So even though technically it is reducing the ploidy to half, but ultimately it is helping in maintenance of the ploidy for the species itself. This is what the basic and main important feature of this particular meiosis. And regarding this, more explanation I gave in the regular lecture. Those who want more explanation, please go through the regular lecture. And next one is regarding the key features of the meiosis. In NCERT they gave all these 4 features. Please observe very carefully. Meiosis, it is having 2 parts like meiosis 1 and meiosis 2. But in the case of mitosis, there is only one. And that too, here the important point is, in the case of meiosis, even though meiosis 1 and meiosis 2 are present, DNA replication occurs only one, that is in the S phase. And this is the similarity with mitosis. Even in the mitosis also, DNA replication occurs only one, that too in S phase only. Next one, meiosis 1 starts after the S phase. Very, very important sentence. Many students are not reading this sentence properly. Please read the NCERT sentence carefully. Meiosis 1 initiates after parental chromosomes have duplicated to form sister chromatids in the S phase. This is what the sentence of NCERT. That means what? Meiosis 1 starts after S phase. That means which one is missing? G2 phase is missing. Why they are saying like this? Because in majority, I am not saying always, in majority of the cases, G2 phase is absent. In some very rare cases, G2 phase is present, but it is reduced. Please understand this. This is what the original concept. But in the NCERT, directly they said that Meiosis I will start after S phase. This is what the meaning of second point. Please read NCERT carefully. That means as per NCERT, if you say G2 is absent, so stick to that particular sentence only. So G2 is absent. That means Meiosis I will occur after S phase only. Next one, pairing of homologous chromosomes will occur and recombination also is occurring. That means crossing over is occurring. Recombination occurs between non-sister chromatids of homologous chromosomes. And this pairing, crossing over concept, all these we are observing only in the meiosis. It is not occurring in mitosis. Next one, 4 haploid cells are formed at the end of complete meiosis. That is the meiosis II. But in the case of mitosis, 2 diploid cells are produced. Suppose in mitosis, meiosis, in both, let us start with diploid cells only. In the mitosis, diploid daughter cells are produced, but in meiosis, haploid daughter cells will be produced. That's what they are saying here. And how many? In the case of mitosis, 2 daughter cells, but in meiosis, 4 daughter cells are produced. That is also one more important point. So this is what. And then we will talk about the complete meiosis. Please remember, this is entirely called as one meiosis. Some students, what they are thinking, that meiosis 1 is one meiosis and meiosis 2 is another meiosis. No, that is wrong. Completely it is called as one meiosis. Sometimes in the numerical questions they will ask you, in one meiosis, I will ask you, anyhow, separately I kept a slide, there I will tell you the calculation part. So now I will ask the question, in one meiosis, how many meiotic apparatus are formed? Now this is the question. Then how you will calculate? Separately I gave the calculation, there I will tell you. So remember this. So that's why completely it is called as one meiosis. Now here also there is an interphase, but in this interphase G2 is absent. That's why G1 and S are present. After S, meiosis 1 will begin. In the meiosis 1 again, karyokinesis 1, cytokinesis 1. In the karyokinesis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis 1. With this, meiosis 1 is completed. And between meiosis 1 and meiosis 2, there is a short phase that is called as enterokinesis. And after this, meiosis 2 will begin. In the meiosis 2 also, karyokinesis 2 and the cytokinesis 2 is there. Within karyokinesis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. So that's why, what is the overall sequence here? G1, yes, prophase 1. Within the prophase 1 again 5 substages are there. Leptotin, zygotin, pakitin, diplotin, diakinesis. After that, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. Then enterokinesis, then prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2. This is the complete sequence for one meiosis. This sequence also you need to remember. So followed by, follows, preceded by, precedes. These questions are compulsory. You need to understand these meanings. Next one, meiosis 1. Now let us go with directly with prophase. Why we are going with prophase? Because the G1 and S phases are almost like mitosis only. That's why we are not giving special interest on that one. Directly we will go with meiosis 1. In the meiosis 1, first we are going with prophase 1. This is a little bit comparatively longer because it is having again 5 substages. It is a little bit longer and typically more complex. Because what is the overall aim of this prophase 1? Let us understand. Homologous chromosomes should undergo pairing, recombination should occur, and then they should separate. Understood or not? Why it is little bit longer? Because here some complex processes are taking place. And what is the overall summary of this? Homologous chromosomes should undergo pairing and then crossing over should occur and then they should separate. This is the overall idea of this particular one. And these are the 5 substages. So now let us first go with the leptotin. Since it is part of the prophase. Please remember, in the entire prophase, condensation is a continuous process. Condensation is a continuous process. And maximum condensation we are observing in diakinesis. In NCERT, they used a sentence called fully condensed chromosomes are seen in diakinesis. This is the sentence. So you need to observe. In the mitosis, where fully condensed chromosomes are seen? In the metaphase. But here, diakinesis. Here diakinesis. In the meiosis 2, if you are talking again, metaphase 2 only will come. So please understand all these sentences. It is not that you are simply by-hearting this. You need to understand this. Now let us go with the leptotin. It is the first phase. So here condensation will begin. Condensation of chromosomes is beginning means what? You can start visualizing these under the microscope. Clear. Next one, in the zygote what happens? Homologous chromosomes will undergo pairing, and this pairing is called as a synapsis. So now here the homologous chromosome should attach to each other. So some proteins will come there and they are helping in attachment, and this complex is called as synaptonemal complex. Since two chromosomes are attached and each one is a dyad, this is dyad, this is dyad, all these are attached. So overall how many chromatids? 4 chromatids. That's why tetrad condition. Overall how many chromosomes? 2 chromosomes. That's why it is called bivalent. So that's why bivalent or tetrad condition. Tetrad is a condition, not a tetrad chromosome. Bivalent is a condition. I will tell you all these things. Observe carefully. So bivalent or tetrad condition is seen first in the zygote. It is first seen in the zygote. The same bivalents are present in zygote, packete, diplote, diakinesis, metaphase 1 also. In all these things bivalents are present, but they are first formed in zygote and they are clearly visible in next phase. What is the next phase? That is the packete. And what exactly happens in the packete? Now these bivalents or tetrads are clearly visible, and here what happens is some group of proteins will come there, and now they are helping in the recombination process. So since group of proteins are attached at one particular location, there you can see some bulged appearance, bulged nodule like appearance. That's why it is called a recombination nodule, and that is where exactly the crossing over or recombination will take place. And this recombination will occur between non-sister chromatids of homologous chromosomes, and this exchange is simply called as crossing over or recombination. It is an enzyme mediated process, and the enzyme involved is called as recombinase enzyme. This much of information is enough. In higher classes you will study about this, and it is a very complex process. Clear? Next one is regarding the diplotin. In the diplotin what happens? Already in packete crossing over completed. Now they should start separate, but for the separation there is a problem because synaptonemal complex is already keeping homologous chromosomes attached. Now first synaptonemal complex should dissolve. So in the diplotin what happens? Dissolution of synaptonemal complex will occur, so that now homologous chromosomes can start separation. I am not saying completely separated, they are start separated. So during this separation, now what happens here? At some locations wherever there is a crossing over, there still you can see X-shaped structures. Wherever there is no crossing over, there they got separated. But wherever there is a crossing over, still they are attached. So you can see X-shaped structures, and these X-shaped structures are called as a chiasmata. And here they are saying specifically one sentence for oocytes of vertebrates, not for spermatocytes. Please observe carefully. Not for spermatogenesis process. In the oogenesis process, please understand this. This sentence once again I link up with human, because human is also part of vertebrates. In the human also this sentence is there. Separately at the end of this one I will tell you for human. So this is though the chapter is very simple, it is having so much of interlinking concepts. So that's why I am telling you that lecture length may be little bit extended, but observe each and every point carefully. In the oocytes of some vertebrates, diplotin can last for some months or years based on the organism. Based on the organism, it is extended for several months or years also. And this sentence is applicable even for human also. That's why in the case of human, oogenesis process is beginning in the fetal stage. Already human reproduction chapter is completed. So that's why you can understand this. It is starting in oogenesis in the case of human is starting in fetal stage, and it is arrested there till the puberty. That means for several years it is arrested. So this concept again you need to interlink with human. Next one is regarding the diakinesis. Now here what happens? All the intermediate chiasmata will be dissolved, and the homologous chromosomes are attached only at the ends. So chiasmata are seen only at the ends, at the terminals. So terminalization of chiasmata, only at the terminals chiasmata are seen. What is the meaning of this? Though chiasmata is present at the ends and though it is removed in between, still homologous chromosomes are attached. That means still you need to use bivalent or tetrad name only. They are not completely separated. So that's what you need to see here. And most important point is fully condensed chromosomes are seen in the diakinesis. Fully condensed chromosomes, direct NCERT sentence. And since it is the last part of the prophase 1, nuclear membrane breakdown, endoplasmic reticulum breakdowns, Golgi complex breakdowns, and the nucleolus disappears. That's it. These are some important points you need to remember. And this is like a transition to the next one, metaphase 1. In the metaphase 1, what happens here? Bivalent chromosome. Please remember, in the diakinase still they are attached at the ends, the same will come and align here. So here, like this, they have given the diagram. So observe carefully. So here what happens? Bivalents are aligned here. Bivalents are aligned at the metaphase plate. So you can also call it as, so here 2 lines of centromeres you can see. That's why generally it is called as biphasic metaphase plate. Biphasic metaphase plate, or 2 equatorial plates also sometimes they are calling. So please observe this particular concept. Now here pairs of chromosomes are aligned. In the normal mitosis of, in the normal mitosis metaphase, individual chromosomes are aligned. All the 46 are present side by side. But here they are present as pairs. 23 pairs of chromosomes are present here. Now they are aligned at the center of spindle apparatus technically. Now this is what happening here. Now here also, in the prophase, by the end of prophase 1, what happens? I forgot to tell you. So by the end of the prophase 1, what happens? So centrosomes will move to opposite poles, spindle poles will be formed, spindle fibers will arise, but spindle fibers will attach to kinetochores and the chromosomes will be aligned at the center of the cell in metaphase 1. Clear? Next one is anaphase. And in the anaphase, what happens? Centromere will not split. Simply homologous chromosomes will move to opposite poles. That means sister chromatids are going to same poles. Names are very important. I gave clear-cut explanation in the case of regular classes. So please go through that if you want more details and more explanation. Okay, this is what happening here. Now towards, with respect to human, if you are talking, 23 are going on one side, 23 are going on the other side. That's it. Next one is regarding telophase. Now here, decondensation will occur. And then around each group, now nuclear membrane will be formed. That means 2 nuclei will be formed here. This is what happening. And here, now it is almost opposite to the prophase 1. Clear? And next one is regarding the cytokinesis 1. So if it is plant cell, different mechanism we studied. If it is animal cell, different mechanism we studied. So finally cytokinesis will occur and the 2 cells will be produced. And what is the ploidy of these cells? Haploid. Please understand, parent cell is diploid and the daughter cells are haploid cells. 2 haploid cells are produced. That's why it is called as a dyad of cells. 2 haploid cells, dyad of cells. That means in the meiosis, actually in meiosis 1 itself, ploidy reduction is taking place. That's why it is called a reduction division. In the case of meiosis, ploidy is reduced to half. That's why it is called a reduction division. And that reduction is actually occurring in meiosis 1 itself. That's why meiosis 2 will resemble the mitosis process. So finally 4 haploid cells are produced. Before going to that, now let us see the NCERT diagram. In the NCERT, they have given diagram for prophase 1, metaphase 1, anaphase 1, telophase 1. What is the problem with NCERT diagram? Observe carefully. In the metaphase 1 diagram, observe this. These are homologous chromosomes. In NCERT diagram, they kept them separately. This is wrong. This is wrong. You should not keep them separately. Actually, these 2 homologous should be attached. These 2 homologous should be attached. In the case of metaphase 1, bivalents are aligned. Bivalents are aligned means they should be attached. Here they kept separately. This is wrong representation. This is one thing. And here it is okay. And in the telophase, what they did is they have already separated the cells. In the telophase, you should not separate. In the single cell, you need to show both the nuclei. After cytokinesis, you need to show them separated cells, right? So this is one small thing that you can easily understand. Clear? With this, meiosis 1 is completed. Before going to the meiosis 2, we need to talk about interkinesis. What exactly happens in the interkinesis? There is no DNA replication, but centrosome duplication will occur. Under the diagrams, once again I will give clear-cut explanation. Centrosome duplication will occur in this. And then it will move to the meiosis 2. And this meiosis 2 almost resembles to mitosis. So quickly we can go through this. So prophase 2, in the prophase 2 once again, condensation of this particular DNA will occur. And then nuclear membrane disappears, endoplasmic reticulum Golgi complex disappears, and that nucleolus also disappears. Okay, this is like mitosis only. Next, metaphase 2. Here at the center of the cell, these chromosomes will be, center of the cell means center of the spindle apparatus, the chromosomes will be aligned. It is almost like mitosis. I don't want to repeat all the points again. Anaphase 2, splitting of centromere, sister chromatids will move to opposite poles. In the telophase, around each group again, nuclear membrane will be formed, ERGC will be formed, and everything will happen. Cytokinesis 2, and here cytokinesis will occur so that now the distribution of organelles and nucleus into the daughter cells will occur. Finally, what happens? Here, now here at the end of meiosis 1, 2 daughter cells are produced. Now each one is individually undergoing meiosis II. So overall what happens? Total 4 haploid cells are produced. 4 haploid means a tetrad of cells, a tetra 4 add means with respect to haploid. So 4 haploid cells are produced at the end of meiosis II. This is what happening. Now in this also there are two types. This was not mentioned in NCERT, and in some cases what happens is, so at the end of meiosis I, 2 individual cells are produced. Again they are individually undergoing meiosis II, so that 4 cells are produced. If this is happening, it is called as a successive type of cell division. Okay, next one, meiosis with respect to meiosis. And in some cases what happens is, at the end of meiosis I, cytokinesis 1 does not occur. After karyokinesis 1, 2 nuclei are produced, but cytokinesis 1 does not occur. Directly it will go into meiosis II. That means again from each nucleus again 2 more nuclei will occur. So overall 4 nuclei will be there, and at the end of meiosis II directly at a time cytokinesis will occur and all the 4 cells will be produced. And this type of division is called as simultaneous type. And the successive type generally seen in monocots, simultaneous type is generally seen in the dicots. But this information was not given in NCERT. If you can remember, that will be better. That's it. Significance of meiosis. Though technically it is reducing the ploidy to off, upon fertilization of the gametes again, the ploidy of the species will be maintained, and this is what the important point. And next one is regarding the recombinations. Because of this crossing over or recombination, some new combinations will be produced in the next generations, so that they are very important for the evolution. Yes or no? Next one. So with this, NCERT content is completed. Now we are going with some other useful and interlinked information. So now also you should be little bit more careful. So observe carefully. Mitotic divisions and generations. This is with respect to calculation. So quickly I will tell you. So observe carefully. What is this one? Suppose this is a single cell, and this single cell is undergoing a mitotic division. With respect to mitosis we are studying. This is the division number 1. Upon one division what happens? 2 cells will be produced. So after one division 2 cells are produced. Now this is one division. Now this will be the second division. Now from the second 2 cells are produced. After 2 divisions how many cells are produced? 1, 2 and 3. After one division, this point is very important. Many students are making mistake here. After so and so division, after so and so divisions, how many cells are produced? Let us see. After one division 2 cells are produced. Yes or no? Now this is one division, this is one division. Now how many cells? 1, 2, 3. That means after 2 divisions how many cells are present? 3 cells are present. Try to understand the relation here. Now suppose this cell is also undergoing the division. Now 1, 2, 3. From this again 2 cells. After 3 divisions how many cells are there? 4 cells are there. After 3 divisions 4 cells are there. Next one. Now again this cell will divide and form 2. And what is the number? 4. After 4 divisions how many cells? 1, 2, 3, 4, 5. You need to understand the relation. After 4 divisions 5 cells are there. Don't worry, though it takes time, just only, but it is very important concept. After 5 divisions how many cells? 1, 2, 3, 4, 5, 6. So you need to understand the relation. After 5 divisions 6 cells. So after 6 divisions 7 cells. After 7 divisions 8 cells will be there. Understood or not? So this is what the relation. Directly I will write this. This is the 6th division, so 7 cells. After 7th division 8 cells are there. Now again, now here 8th division, 9th division, 10th division, 11, 12, 13, 14, 15. So after 15 divisions how many cells will be there? 16 cells will be present. So that formula you need to observe very carefully. You don't need to by heart, understand this. After n number of divisions how many cells will be there? N plus 1 will be there. After n number of mitotic divisions n plus 1 cells will be formed. Next point, next question. If you want x number of cells, from initial single cell, how many mitotic divisions are required? X minus 1 mitotic divisions are required. So questions can be asked in any of these ways. So please observe this concept carefully. Clear? This is with respect to number of mitotic divisions. Now we will talk about number of mitotic generations. This is the 0 generation. This is the initial cell, right? This is the 0 generation, and this is the first generation, and this is the second generation, and this is the third generation, and this is the fourth generation. You can go keep on like this. And in this generation means like this you need to count. In the 0 generation, how many cells? 2 cells. How many cells? 2 cells. In the first generation, total how many cells are there? 1, 2. 2 cells are there. In the second generation, 1, 2, 3, 4. 4 cells are present. Yes or no? Next, in third generation, 1, 2, 3, 4, 5, 6, 7, 8. 8 cells are present. And in the fourth generation, total how many are there? 16 cells are present. Observe carefully. 0 generation, 2 cells. Of first generation, 2 cells. Second generation, 4 cells. Can you find any relation? Yes, it is the 2 to the power n. And n indicates generation. Yes or no? So 2 power 0, 0 generation, 2 power 0. Yes or no? It is the single cell, only a single, sorry, it is the one cell. 2 to the power 0, anything power 0 is 1, right? So it is 2 to the power 0, 1 cell. But it is 2 to the power 1, first generation. 2 to the power 1 will be what? 2 cells. Second generation, 2 square, 2 to the power 2, it will be 4. Third generation, 2 cube, so it is 2 to the power 3, it is 8 cells. Fourth generation, 2 to the power 4, 16. Suppose they are asking for fifth generation. Fifth generation, then what you will write? 2 to the power 5, how many? 32 cells will be there. So in this way, generation means this is how you need to calculate. Individual mitotic divisions means individually you need to count. Understood or not? Now the question is, for 32 cells, from initial single cell, how many mitotic divisions are required? How many mitotic divisions are required? 31 mitotic divisions are required. How many mitotic generations are required? 5 mitotic generations are required. This you need to observe carefully. In this way, if they are asking for 2, 4, 8, 16, 32, you can write, no problem. Now I will give the last example on this. Now let us see. Suppose you want 20 cells. You want 20 cells. How many mitotic divisions are required? How many mitotic generations are required? Now tell me the answer. If you want 20 cells, how many mitotic divisions are required? It will be -1. So 19 mitotic divisions are required. So what is the answer? 19 mitotic divisions are required. But if they are asking for generations, so directly 20 you cannot get anywhere. So what you need to do is, if it is 4 generations, only 16 you will get. So once it is going beyond this, you need to go for the next complete generation. There is no like 4.2 generation, 4.3 generation. It is not like this. It should be a whole number. Either it should be 4 or 5, but 4 will give less number. So you need to go for 5. Even though some extra are there, it does not mind. So you want 20 means compulsorily it should go for 5 generations. So it is the 5 generations. Understood or not? In this way, you need to go for the calculations related to this type of questions. I hope you understood this. If you did not understand this, no problem. So I gave more examples and everything I gave in the regular lecture, because this is a revision, so I cannot give more time for this. But I am trying my best to explain all these things. Clear? So if you want more details, please go through the regular lecture. So with this concept is completed. Next one. So how to calculate the number of meiotic divisions? This is for meiotic divisions, right? Number of meiotic divisions required for male gametes formation, female gametes formation, zygotes formation, all the things. This is the basic concept, and now we will focus on this particular one. What is the basic information here? With respect to meiosis, if you are observing, in the case of meiosis, what happens? From the single meiosis, how many cells are produced? 4 cells are produced. In one meiosis, this is completely one meiosis. Don't consider 2 meiosis. This is one meiosis. This is what I am saying. One meiosis will form 4 cells. Nice. So these 4 may be male gametes, these 4 may be female gametes. Nice. But here there is one technical problem. If it is about the male gametes, one meiosis will form 4 male gametes, because in the case of male gametes, all the 4 are functional. So one meiosis will form 4 male gametes. And if you are talking about the female gametes, in the case of female, what happens is, even though let us consider that all the 4 are produced and 3 are not functional, only 1 is functional. So in the case of female, what happens? One meiosis will produce only one female gamete, one ovum, one egg cell, whatever it is. So once again I am repeating, in the case of male gametes, one meiosis will produce 4 male gametes, but in the case of female, one meiosis will result in only one female gamete. So that's why for the calculation purpose you will get the difference. Now we will see one small example. Let us take small example. Suppose 12 male gametes, 12 male gametes. Sorry, first I will tell you the basic concept before directly going with the question, because you may be confused. See, one male gamete fuses with one female gamete, okay. Please remember, simply I am using male gamete, female gamete. You can use the terms. For male gamete, you can use the term called as sperms. For female gamete, in the case of plants, generally we are using a term called egg. In the case of animals, generally we are using the term called as ovum. Anything you can use, simply basic concept I am saying. So these two will combinedly, they will form one zygote. These two will form one zygote. In the case of animal, simply we are saying one zygote, and in the case of plant, you are saying one seed. Okay. This is what generally they are asking some questions. So in this way, we are talking about this particular information. Now let us focus on this. So for one male gamete here, one female gamete, so that combined they are forming one zygote. That means for one zygote or one seed formation, you need one male and one female gametes. This is the basic information. Based on this, now let us write about some questions. Suppose you want 12 zygotes. Directly I am using the name called 12 zygotes. For the formation of 12 zygotes, during the gametogenesis process, how many meiotic divisions, meiosis process are required? This is what the question is. How you will calculate? First we will see for the male. This is the symbol for male, this is the symbol for female. So here what we need to say here, 12 male gametes are required and 12 female gametes are required. This is the basic logic. For 12 male gametes, what we studied, one meiosis will form 4 male gametes. So for 12 male gametes, how many are required? 3 meiosis are required. So here what is the answer? I am writing in a different color. So here the point is 3 meiotic divisions are required. 3 meiotic divisions are required on this side. But here what we studied? What is the concept we studied? In female, one meiosis will form one female gamete. But here you want 12 female gametes. For 12 female gametes, how many meiosis are required? 12 meiosis are required. 12 meiosis are required. So what is the total number here? 12 plus 3, it is 15 meiosis are required. 15 meiosis are required. This is how you need to do the calculation. Understood? Now for the same thing, I will try to give a particular formula. What is the formula here? Suppose you want n number of zygotes. If you want n number of zygotes, for the female gametes, how many meiotic divisions are required? m number of meiotic divisions are required for female gametes. But for the male gametes, how many it will be? It is 12 by 4, it will be 3. Yes or no? So it is n by 4. So now what is the overall number here? 15, how you are getting? N plus n by 4. So for so and so number of zygotes, if they are asking, how many number of meiotic divisions are required? N plus n by 4 are required. Same thing you can also write as n into, take the n as common. So here it will be 1 plus 1 by 4. Yes or no? N plus n by 4 you can write, or n into 1 plus 1 by 4 also you can write. So that how you can write this one? N into, take the LCM for this, it is the 4. So here you will get 4 plus 1. Please remember this, same information they can represent in different ways. You need to know whether it is true or false. So how you can write this one? So it will be what? 5 by 4 n. Can you write like this or not? Yes, you can write. Yes, you can write. Don't think that this is the only right answer. Because I saw some faculties, they are thinking that this is the only right answer. Please remember, if you know mathematics, you can represent in any of these forms. So please understand this. Okay. So you can write like this also. Any of these are correct. So these are the number. Understood or not? This is how you need to calculate how many meiotic divisions are required for male gametes, for female gametes, for so and so number of zygotes. Clear? So suppose it is like a normal complete number, so you can easily get this number. Okay. Instead of 12, suppose I am asking 13, then what you will do? Now I want 13 zygotes. Now do the calculation and tell me the answer. Now directly I am giving the answer. I am not waiting for your answer. If you want to do it, pause the video and then do it and then continue it. So here you want 13 male gametes, 13 female gametes. 13 male and female gametes you want. Now, for 13 female gametes, how many meiotic divisions? Here 13 divisions are required. But for the male gametes, you just observe carefully. 1 division, 4, 2 divisions, 8, 3 divisions, 12, 12, 4 divisions, 16. But there is no like intermediate like this. Once it is crossing 12, but you need more than this, right? So you need to go for the complete 4. There is not like 4.1, 3.1, 3.2 like this. You should not write. You need to go for the next complete one. So here 4 divisions. 4 divisions. Because of 4 divisions, what happens? 16 are produced. Even though 16 are produced, you want how many? 13 only. Use 13. So how many will be wasted? 3 will be wasted. There are some questions in MCQ books. They are asking that how many pollen grains will be wasted. How you will calculate this? How many are required? 13 are required here. So remaining 3 will be wasted. They are not used. Wasted means they are not used. But still you need to go for the 4 divisions. 4 divisions are required. 4 here, 13 here. So add this. So 13 plus 4. So it will be 17 divisions are required. Suppose you want to use the formula in this case. What you will do? Suppose this is n. What I said? 5 by 4 n I said. Yes or no? Or n plus n by 4, anything you can use. No problem for this. So what you will do here? 5 by 4 into n is what? 30. So what happens here? 4 3s are. And 1 is there. So dot 10. So it will be 2, 5. Like this you are getting. Yes or no? Once you are getting like this, you need to go for the next complete number. Okay. So here you should not go. First multiply and then you should go for the next complete number. So when you are simply multiplying like this, now what happens here? So or else the same thing. Don't get confused. You will get exactly the same answer. Some students have a habit of calculating like this. So n plus. So here it will be 13 plus. It is 13 by 4. Now it will be what? 3.25. Now it will be what? 16.25 you are getting. Yes or no? So here also you will get the same thing. 16.25 you are getting. Anyhow you will get exactly the same number you are getting. So now what you need to do is, when you are getting 0.25 extra, so it is more than 16. So in that case you need to go for the next complete number, that is 17. You need to go for the complete whole number, right? So 17. So here also you are getting the 17. This is the answer. So in this way, if you are using the formula, you are getting point something like this, then you need to go for the next whole number. Please understand. This is how you need to solve these particular questions. Clear? This is how we are doing this type of numerical questions. Next one. This is for this particular concept. Next one. Number of meiotic apparatus. Very good. This is also one important concept. Number they are asking. So you need to understand this concept. What is this particular concept with respect to number? First, let us talk about the mitosis. Then we will go with the meiosis. What is this mitosis concept? In the mitosis, what happens? This is for the single cell, right? And now it will form two daughter cells. Suppose it is 2n. This is also 2n, 2n. Nice. But in this parent cell only, mitotic apparatus. This is mitotic apparatus. Spindle apparatus or mitotic apparatus is formed. What is one mitotic apparatus? I told you already. What is one mitotic apparatus? Two spindle poles plus spindle fibers combined. So this is spindle pole with spindle fibers. Okay. Spindle pole with spindle fibers. Completely it is called as one apparatus. That's it. So one mitosis. In one mitosis, how many mitotic apparatus? One mitotic apparatus or one spindle apparatus. In one mitosis, one mitotic apparatus or one spindle apparatus. This is the calculation. Suppose you are going for meiosis. This is very important. Observe carefully. In the meiosis, meiotic apparatus or spindle apparatus there also. Here what you will say? Parental diploid cell. After meiosis 1, 2 haploid cells are produced. Yes or no? And again they are undergoing meiosis 2. So that 4 haploid cells are produced. Where the apparatus will be formed here. During meiosis 1, in this parent cell, spindle apparatus will be formed. This is the spindle pole with spindle fibers. Completely it is called as one spindle apparatus. After meiosis I, two cells are produced. Now during meiosis II, spindle apparatus is formed in these cells, not in these cells. Not in these cells, in these cells, in these cells. Please remember, generally the orientation of this spindle apparatus and in the daughter cells it is 90 degrees. 90 degrees it is, but for your understanding purpose I am writing vertical only, so observe carefully. Now this is one spindle apparatus in one cell, and here also there is one spindle apparatus. Okay, one spindle apparatus in this. Now what happens? Now overall how many? In this it is 1, in this it is 1, in this it is 1. So 1 plus 2, total how many? 3. So in one meiosis, how many spindle apparati? 3 spindle apparati or apparatuses are formed. Please understand this. One meiosis, 3 spindle apparatus. One mitosis, 1 spindle apparatus. This is the question. Now the thing is here. That question can be asked here. In this case, for the 12 zygotes, now during the formation of these 12 zygotes, okay, from during the gametogenesis process, total how many meiotic apparati are produced? Tell me. How we will calculate? We have already calculated the number of meiosis. 15 meiosis is the answer you got. In one meiosis, 3 spindle apparatus. 15 means 15 into 345. Understood or not? So here also you got some number, right? Multiply with the 3. In this way, questions and concepts can be interlinked. So that concept you need to understand. That is also part of NCERT. They can ask the questions, right? This is what about the number of the spindle apparatus. Nice. Next one. Ovum formation in human female and the important stage. This is also another very important concept. This you are studying under human reproduction chapter, but still it is connected with cell cycle, cell division. Quickly I will tell you, try to understand this. Regarding this, I gave complete detailed information in cell cycle, cell division chapter itself with respect to human reproduction, mainly with respect to this oogenesis process. Very clearly I have explained in regular chapter. So you please refer that particular video once again, because this is a very important concept. Now let us see. For the human gametogenesis process, first we are talking about the spermatogenesis process. Spermatogonia, these are diploid cells, and they are dividing by mitosis, not meiosis. By mitosis they are undergoing division, so that several cells are produced, and out of this some of them will undergo differentiation to develop into primary spermatocytes, which are also diploid cells. And now these cells will start undergoing the meiosis. So first meiotic division will occur, so that haploid secondary spermatocytes are produced. And again they are undergoing second meiotic division, so that again they are producing haploid spermatids. And these haploid spermatids, they are undergoing differentiation to form haploid spermatozoa. In the case of spermatogenesis process, from one primary spermatocyte, 4 spermatozoa are produced. Clear? And it is a continuous process. Once it starts, it is a continuous process. It is not stopping in between. Clear? For this there is no problem. Clear? Come to this oogonia process, oogenesis process. In the oogenesis process, the initial cells are called as oogonia. These are also dividing by mitosis, not meiosis. And here also they are producing many same type of cells. And out of them, some of them are undergoing differentiation to develop into primary oocyte. And this is also diploid cell. Primary oocyte is also diploid cell. Now it is undergoing the first meiotic division. But though it is undergoing first meiotic division, and here it is starting in the fetal life, but it is arrested. Where it is arrested? It is arrested in diplotin. Already in the diplotin we studied, no? In many vertebrates, in most of the vertebrates, diplotin stage is arrested for several months or years, right? Now this is years in the case of human. It is arrested. Where it is arrested? In the diplotin phase of prophase 1 of meiosis 1. It is arrested. It is stopped. In the fetal life it is started. Which one is started? Oogenesis process is started. Meiosis 1 is initiated, but it is stopped in diplotin of prophase 1. And there it is stopped. And then what happens? After the completion of 9 months, now the birth of the baby is taking place, and then the baby is growing, and in the during the puberty, when the menstrual cycle starts. Now, puberty they gave, no? Now here when the menstrual cycle starts, in each menstrual cycle some cells will continue further. Some cells will continue further, means after diplotin they will enter into diakinesis, metaphase 1, like that they are continuing. And what about the remaining cells? They are still present in the diplotin only. So in this way, in every menstrual cycle, some cells will continue further. Remaining are still present in diplotin only. What are these problems and all, that we will discuss separately in the genetics chapter. Okay. Now let us continue here. Now here, in each menstrual cycle, some cells will continue further. Now from the diplotin, now they are continuing into diakinesis, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. Once it is completed, what happens? Secondary oocyte is produced. And along with this secondary oocyte, there is a first polar body. Out of the two cells, one is functional, other one is not functional. Non-functional is first polar body. Other one is functional, it is called as secondary oocyte. And this first polar body may or may not undergo second meiotic division. It depends on the species. And this secondary oocyte will start undergoing second meiotic division. But here also it is once again arrested. Where it is arrested? It is going into prophase 2, metaphase 2, and metaphase 2 is arrested. So this secondary oocyte which is arrested in the metaphase 2, now that will be ovulated from the Graafian follicle. From the Graafian follicle, whatever the ovum you are saying, but technically it is not a ovum, it is a secondary oocyte that was arrested in metaphase 2, that is ovulated. Now that is entering into the fallopian tube. Now it is moving in the fallopian tube. And when that secondary oocyte arrested in metaphase 2 is present in the ampulla region, if sperm cells are reaching that particular location at that time, then fertilization may occur. If fertilization occurs, please understand, fertilization means what? When sperm cell enters into that secondary oocyte, then only it will move further from metaphase 2 to anaphase 2 and it will complete. Then only functional ovum will be produced and then second polar body will be released. And after this, only fusion of male and female nucleus will occur. Please understand all these things, and these details more clearly we are covering under human reproduction chapter. But please understand this. What are important here? During the fetal life, primary oocyte is undergoing the meiosis 1, but it is arrested in diplotin of prophase 1. That is one important point. And during ovulation, secondary oocyte arrested in metaphase 2 is actually released. This is another important point. Many students are making mistakes for this. Please don't do mistakes. These are with respect to cell cycle phases. You need to know the ploidy of each and everything, and in which stage it is started, where it is arrested, everything is very important for examination. More details I gave in the regular lecture, so if you are interested, so please refer to that particular lecture. Next one, some important names. Eccentric. If centrioles are not involved, then it is called eccentric division. So we are observing in the plant cells. Anastral, asters are not present. This is also for the plant cell. What is opposite? Centric, astral. Astral also called as amphiastral. Both the asters are there. So centric, astral or amphiastral terms. These we are using for animal cells. Eccentric or anastral mitosis we are observing for plant cell. Clear? Next one. Regarding the types of meiosis, not mentioned in NCERT, but important to know. Let us quickly go through this. Zygotic meiosis. In zygotic meiosis, what happens? Zygote undergoes meiosis so that haploid cells are produced, and now they are developing into haploid individuals. They will produce haploid gametes. Haploid gametes will undergo fertilization so that they are again forming the zygote. This is called as zygotic meiosis. Next one is called gametic meiosis, because meiosis forms the gametes, and this with respect to human you can apply this. Zygote is developing into diploid individual, and in the diploid individual some cells will undergo meiosis to form haploid gametes. These haploid gametes will undergo fertilization to restore again the zygotic condition, diploid zygote condition. Clear? With respect to human you can apply this. Next one, sporic meiosis. In this what happens is zygote develops into diploid individual, and here meiosis will occur to form the haploid spores, and these haploid spores will develop into haploid individuals. Haploid individuals will produce haploid gametes. Haploid gametes will undergo fertilization to form diploid zygote, and this process will repeat. This is called as sporic meiosis. So these are the different names. Generally in the plant you are studying about these particular names. Clear? Next one, Mitosis. It is also called as a direct cell division. Here what happens is, without the nuclear division, the segregation of chromosomes is taking place within the nucleus itself, and finally nucleus will divide, and this is how a mitosis will occur. Clear? endomitosis. And here also what happens is nucleus will remains like that only. Inside the nucleus this chromosomal separation will everything will occur. This is what the endomitosis. And the endoreduplication generally we are observing in salivary gland cells of some of the insects, and here we are observing the polytene condition. Poly, many, teeny means thread-like structures. Many thread-like structures because of endoreduplication, continuous DNA replication that is occurring inside the cell. And generally these cells are arrested in S phase itself, and then these cells will die. They cannot complete the cell division process. That's it. You don't need further information regarding this. Next one, lamp brush chromosomes. Generally in the oocytes of amphibians, already we studied, no? Diplotin stage is arrested for several months and years. Generally in the case of amphibians, what happens is, so since it is arrested for longer duration, now for that much of duration cell has to survive. So it has to produce some RNA and protein. So generally in the normal condition, what we are saying, if at a time the meiosis completes, then it is okay. But if it is arrested for longer duration, so some proteins are required. So in that case, now the DNA is forming some loops here and there, and these loops finally they are appearing as a lamp brush like structure. So in this way these are called as lamp brush chromosomes, and these are the loops at which actually the active RNA synthesis occurs. If RNA is produced, from that RNA proteins can be produced so that cell can survive. So in this way these loops are actually the locations of transcription. But anyhow, this information is not required, but important is lamp brush chromosomes are seen in oocytes of most of the vertebrates, mainly in the case of amphibians, but these lamp brush chromosomes are not seen in the mammals. So in the case of human, even though those that in the oogenesis process, though it is arrested in the diplotin, lamp brush condition is not seen in the case of human. Mainly generally they will give the example of the frog. So please remember this, because frog is an amphibian. So there you are observing this one. So please remember this. Next one. So mainly in the case of the genetics concept, so we are studying about the gametes formation process. Suppose you are starting with capital T small t condition. Ultimately the chromosomes are distributed into the gametes, so that 50% are having capital T, 50% are having small t. So this you can easily explain here. And next one is regarding capital R smaller, capital Y small y condition that is producing 4 types of gametes. Capital R capital Y, capital R small y, small capital Y, small y, all of them with equal proportion, 1:1:1:1 proportion, right? So this we can explain with the help of random alignment of chromosomes. What is random alignment of chromosomes? During the metaphase 1 and during the metaphase 2, the alignment of chromosomes here is a random process. So the alignment of each pair of chromosomes is independent of other pair of chromosomes. So that's why we are getting various combinations. These combinations are resulting in formation of various types of gametes. So this concept is also explained based on cell cycle concept only. So I gave very clear cut explanation in the regular lectures of cell cycle, cell division. So please go through that. So with this, the basic information of cell cycle, cell division is completed. Interlinking concepts are also completed. And finally we came to this phases and the diagrams. Now here I will not fill it, I will draw the separate diagrams, but I will go a little bit quickly because already we are going a little bit beyond our time. So what I will do is first I will draw for the mitosis, then we will go with meiosis. I will draw the name of the particular phase, I will draw one rough diagram, and then what are the conditions for the chromosomes, Ploidy, C-value, everything we will write. Okay, so better you also try to give the answers, so that let us quickly try to complete this one. First we will start with the mitosis. And before directly starting with this, first let us assume that we are studying for the human. In the case of human, haploid is 23 chromosomes, diploid has 46 chromosomes. So this information anyhow you know. So here what I will do is, within the diagram I will use these names also, numbers also, so that you can understand the concept very clearly. Okay, so this information also I have clearly covered in the regular lectures also, and there I took a little bit more time because that is a regular lecture, so I need to explain it a little bit slowly. But since it is a revision, I am going a little bit quickly. So those who are having any doubt, so you can ask me. So if you are having the access to the app, so please go through the lecture also, so you will get more clarity. But this is very important. Once you understood this table, you can answer any question from this particular chapter. Let us quickly go through this. In the mitosis, first we will go with G1 phase. In the case of G1 phase, a plasma membrane is present and a nuclear membrane is also present. In the diagram also, our main aim is, anyhow in every phase plasma membrane is present. We are studying for animal cell, plasma membrane is outer covering, it is present in all the phases. Our main focus is what is happening to nuclear membrane. And second point is whether it is present as thread-like chromatin or condensed chromosome. That is another point. Next one is centrosome. Centrosome. These are the three important points we are focusing: nuclear membrane, and then chromatin or chromosome, and then centrosome. These are the three things that we are observing. Now let us see. In the G1 phase, nuclear membrane is present, and a single centrosome is present, and chromatin thread-like structures are present. Okay. For understanding purpose, I am making the centrosome. This is how they are present. But since we are studying for human, we are generally starting with diploid cell. So like this, how many are present? 46 are present. 46 are present. So what is this chromosome? Single chromatid. So it is called monad. And it is not attached with homologous chromosome. So it is monovalent. It is monad and monovalent. It is monad, monovalent. What is monovalent and bivalent? If chromosome is present individually, that is called as monovalent. If homologous chromosomes are attached, it is called bivalent. So please understand, in the entire mitosis you will see monovalent only, because there is no pairing of homologous chromosomes. In the entire mitosis, monovalent chromosomes are only present, no bivalent. But how many chromatids are present? If single chromatid, monad. If two chromatids, dyad. This is how we are writing. Next one is regarding the ploidy. And then we will write the C-value. These are the informations we need to write. Monovalent and monad. Clear? Ploidy. What is the ploidy? Single thread like 46 are there. One chromosome. One centromere means one chromosome. One chromosome. Like this, how many? 46. So 46 chromosomes. 46 chromosomes means it is diploid. Clear? Next one. C-value. C-value. One chromatid has one double-stranded DNA. Like this, total how many? 46 are there. So 46 into 1 means 46 DNA molecules. 46 DNA molecules means it is a 2C. In the beginning of the lecture, I told about all these things. So directly we are writing the names, values based on the condition. Clear? This is the G1 phase. Next one is regarding the S phase. S phase. Plasma membrane is present. Nuclear membrane is present. Here also thread-like structures are present. But DNA replication occurs. DNA replication occurs means what? Sister chromatids are produced. So here two chromatids are there in each, and single centromere. Single centromere means it is one chromosome. Like this, how many are there? 46 are present. And regarding the centrosome, duplication occurred. Centrosome duplication occurred. Clear? And here what you will write? It is monovalent. It is monovalent, but it is a dyad chromosome. On single chromosome, two chromatids are there. So it is a dyad chromosome. 46 into 1 centromere. So total 46 chromosomes. So it is diploid. So ploidy remains the same. But what happens to C-value? Now in each chromosome, 2 DNA molecules. 2 into 46 means 92 DNA molecules. Means it is 4C. Amount of DNA is getting doubled. But ploidy remains same. Chromosome number remains same. This is what they have mentioned in NCERT. This is for the S phase. Next one is regarding the G2 phase. In the G2 phase, plasma membrane is present, nuclear membrane is also present. Here also it is a thread-like structure. The diagram almost resembles the S phase only. And here also centrosome is present. Inside some proteins, different proteins are produced, size increases, but our focus is on only these points. So diagram will remain same only. And here it is again monovalent. I think better I will write MV. Monovalent. Every time writing this will take more time, right? Monovalent. And here it is a dyad chromosome. Nice. Next one. What is the ploidy here? Here also 46 chromosomes. So 2N. So here 2 into 46, 92 DNA molecules. So it is a 4C condition. It is a 4C condition. Observe carefully. So in the S and G2, these values will remain the same. Next one. Next one is regarding the prophase. Plasma membrane will remain same only, but what happens to nuclear membrane? Nuclear membrane will start undergoing breakdown. I am using a dotted line. At the end of the prophase, it will disappear. So here let us use the dotted line. Next one. Here until now in the interphase, it is like a thread-like structure. That's why it is a chromatin. But in the prophase, it is undergoing condensation. This condensation is a continuous process. You cannot show this continuous process. So directly I will write this as a condensed form. two chromatids, okay, and this is one chromosome because single centromere. Like this, how many are there? 46 are present. Clear? With respect to centrosome, what you need to say? Movement of centrosome to opposite poles. In the late prophase, they are going to the opposite poles, and now from this spindle fibers are also coming out. Yes or no? In the animal cell, astres are also there. So all this information is there. That's it. Nice. This is what the some basic rough diagram. That's it. And here again monovalent. Two chromatids are there. So it is a dyad chromosome. Clear? Next one. What is the ploidy? 46 into 1. So 46 chromosomes. So it is 2n. And what is the C value here? C value is here 2 DNA molecules into 46. 92 DNA molecules. So it is a 4C. This is the information. So see this, how exactly we are changing the diagram. So here spindle fibers are formed, but they are not attached to kinetochores. They are formed but not attached. When they will attach? In the metaphase they will attach. In the metaphase they will attach. Here plasma membrane is there, but in the metaphase don't draw the nuclear membrane because nuclear membrane disappeared at the end of prophase. So that's why don't draw the nuclear membrane. And this is the spindle pole. Spindle pole. And here what happens? Spindle fibers will attach to chromosomes at the kinetochores, and then they will bring the chromosomes to the center of the spindle apparatus like this. They are present. This is 1, 2. Likewise, all the 46. Please remember they are not pairs. Individual chromosomes present side by side. And then here they are attached. Spindle fibers, they are attached like this. Yes or no? And the kinetochores, they are attached to spindle fibers from opposite poles. In this way, all these are aligned. How many will be there? 46 are there. Clear? Now tell me, here what is the condition here? Here also individual chromosomes. So monovalent. And then each chromosome has two chromatids. So it is a dyad chromosome. Nice. Next one, what is the ploidy here? 46 into 111 centromere. So it's total 46. So it is 2n. And the DNA content also 4C. It remains same. Next one. Next one is regarding anaphase. During anaphase, what happens? Plasma membrane remains same. There is no nuclear membrane, but splitting of centromere will occur. Now observe carefully. This is one spindle pole. This is other spindle pole. Splitting of centromere will occur. So one centromere will split into two. So this is one centromere. This is one centromere. And for this, there are two arms. For this also, two arms are there. Two arms. Centromere is facing towards the pole. Centromere is facing towards the pole. Arms are trailing behind. They are coming behind. So arms are facing the metaphase plate, and centromere is facing towards the poles. So it is going in this direction. It is going to that pole, and it is coming to this pole. Yes or no? And for this one also, it is going to this direction, and it is coming to this direction. In this way, now observe carefully. All the 46 are present side by side. At the center, centromere is splitting. So top 46 will go to one side. So on the top, 46 will go to one side. Bottom 46 will come to other side. Many students are making mistake here. While writing, they are writing 23, 23. That is wrong. At the center, you can split like this. Upper 46 will go on upper side. Bottom 46 will go on bottom side. So observe very carefully. That's what I am saying. Don't try to by heart. Understand how exactly they are changing from one phase to next phase. Then you can easily understand this. Now what happens is, overall you just see, this is one chromosome, individual chromosomes are there, no pairing, right? Individual chromosomes. So it is monovalent. But on one chromosome, how many chromatids are there? Please remember, these are two arms combinedly, it is one chromatid. So 1, everywhere it is one chromatid. So one chromosome with one chromatid is monad chromosome. Here one chromosome with two chromatids, dyad chromosome. But here it is monad chromosome. Now we observe the change. Clear? Now what is the ploidy? One centromere, like this. 46 here, 46 here. So 46 plus 46. In a single cell you need to count. In a single cell total 92 centromeres. 92 centromeres means 92 chromosomes. 92 chromosomes means ploidy doubled. From metaphase to anaphase, ploidy is getting doubled. But what happens to C value? That remains same. One chromatid has one DNA. One double strand DNA. 1. Here 46, here 46, total 92 only. So here C value remains the same. Now observe very carefully. I am going a little bit back, don't get worried. So here we want to compare between G1 and S. For G1 and S, ploidy remains same, but C value is getting doubled. But here what we are saying, from metaphase to anaphase, C value remains same, but ploidy is getting doubled. So that's why don't give the relation between these two. Observe the diagram, observe the phase. According to that, give the values. Don't think that if it is haploid, it is always C only. It is not like that. Don't give that type of conclusions. Clear? Understand the concept. With this anaphase is completed. In the next telophase, what happens? The single chromatids, now they will become again thread-like structures. So observe carefully. Telophase. Telophase. In the telophase, what we are saying? So here on this side, one side, now they are becoming thread-like. On one side, how many are going? 46 are going. On the other side also, 46 are going. Now around this group, there is a nuclear membrane. You can draw the nuclear membrane. Nuclear membrane. And here is one centrosome, and here is one centrosome. Clear? This is what about the telophase. In the telophase, for the complete cell, if you see, total, what is the type of chromosome? It is monovalent. It is monovalent and monad chromosome. Monovalent and monad. And what is the total ploidy? For the complete cell, if you see, what is the ploidy? 46 plus 46, 92. That means it is 4n and the C value is 4C. But at one pole, if they are asking, please remember, if they are simply asking the telophase, you need to write for the complete cell. Generally, they may mention that at one pole, at one side. At one side, if they ask, one side means only information for one side. What you need to write? Monovalent monad. This will remain same. Only for one side, if you see, 46 chromosomes are present, so it is 2n. Only for one side, if you see, what is the C value? It is 2C. 2n, 2C. Yes or no? And then after cytokinesis, now what happens? This will divide, right? So two cells are produced. And here each one is having its own nucleus. Sorry. And here it is the centrosome. Here is the centrosome. And in each one, what happens? Single thread-like structures, 46. Here also single thread-like structures, 46. If you see this diagram, it is exactly same as G1 phase. So this is the G1. After cytokinesis, once again, it is the G1 phase. These are the cells. Now in each cell, let us see in each cell. If you see the each cell, what happens? In each cell, what happens? Monad, monovalent and monad chromosomes. Each cell, if you see, it is the 2n, 2c. Originally, if you see, each parent cell is like this only. Each parent cell is like this only. So in this way, here exactly same like parent cells we are getting in mitosis. This is what they are saying. Understood or not? So that's why in the case of mitosis, even crossing over recombination concept is also not there. So that's why parent and daughter cells are exactly 100%, even with genetic complement also they are same. This is what about the mitosis. So these diagrams one by one, you should write and practice on your own. Then only you will understand this cell cycle, cell division. If you can't write like this, then there is no use in by hearting the NCERT. Okay. Next we are going with the meiosis. Here also we will talk about the human. So 2n is equal to 46. Okay. Let us quickly go with this. And here also we are first starting with the G1 phase. Plasma membrane is present. Nuclear membrane is present. And here single centrosome is present. Single thread-like structures 46 are present. So what is the condition here? Monovalent, monad. And then what is the ploidy here? It is 2n, 2c, 2n, 2c. Directly I am writing here. Okay. Next one is regarding the S phase. In the case of S phase, plasma membrane is present. Nuclear membrane is also present. And the centrosome will undergo duplication. Yes or no? And next one here double thread-like structures are present. Like this 46 are present. So that's why these are monovalent. But two chromatids are there. That's why it is dyad chromosome. So what is the ploidy here? Ploidy remains the same, but the C value is getting doubled because chromatid number increases. So this is what regarding the S phase. Please remember, in the meiosis we are not writing G2 phase because it is absent. Directly we will go with the meiosis one. In the meiosis one, karyokinesis one. Within that first one is prophase one. And in this again 5 substages. So first we will start with leptotin. You need to know about each subphase also. It is not that you will draw single diagram. How you will draw a single diagram for prophase 1? You need to know individual. Leptotin, plasma membrane is present. Here nuclear membrane. Please understand. 5 substages are there. So every time I cannot say that this much broken. So I will use a dotted line. Simply understand that in the prophase 1 it is undergoing nuclear membrane breakdown. That's it. Nuclear membrane breakdown. So I am using a dotted line here. Okay. Centrosome, what I will do is, for individual substage I am not moving. It is slowly, it is moving. Leptotin, zygotin, packetin, diplotin, diagenesis, right? Slowly it is moving, and finally in the diagenesis what happens? It is present at opposite sides, spindle fibers will come. All these things are happening. But slowly we cannot show that. So what I will do is, for leptotin, zygotin, packetin, and then diplotin I will keep here only. Finally in the diagenesis I will show on opposite sides. That will be better. Okay. Okay. This is for. And next one, regarding the chromatin. Now this is the prophase 1. So it is start undergoing condensation. So what you need to do? You need to make like short condenser structures. And this condensation is also a continuous process. So this continuous process we cannot show in the diagram like this. So directly I will write like this one. So these are individual. Please remember here individually all the 46 are present. All the 46 are present individually like this. Don't make any pairs here because this is leptotin. In the zygotin they will undergo pairing. All the 46 are present individually. You can start visualizing them under microscope. These are monovalent but dyad chromosomes. Clear? 46 chromosomes are present, so it is 2n, and the C value is 4C. This is for leptotin. Then we are going with zygotin. In the case of zygotin, plasma membrane is present, nuclear membrane is undergoing breakdown, and these centrosomes, already I said no. So slowly it is keep on moving to opposite side. Nice. And these things what happen? This is very important. Here what we are doing is pairing of homologous chromosomes will occur. Observe carefully. Suppose this is chromosome 1, this is also chromosome 1. Suppose this is chromosome number 2, it will pair with chromosome number 2. So in this way what happens? 23 pairs will be present. 23 pairs will be present. Pairs are present. Even though 23 pairs are present, individually if you count centromeres, what is the total number? That is nothing but 46 chromosomes only. Here also number of chromosomes is 46 only, but they are present as pairs. Previously they are not present as pairs, they are present as individual 46. Here also 46 are there, but they are present as pairs. This is very important point. Clear? Now let us see. Here what happened? Here they are not present. Here individual chromosomes, that's why monovalent. But here chromosomes are attached. It is a condition. You cannot say bivalent, you cannot say bivalent chromosome. You cannot use the term called bivalent chromosome, bivalent chromosomes. It is a condition. In this condition, we are observing two chromosomes together. So that's why it is a bivalent condition. Bivalent chromosomal condition, bivalent chromosomes. It is not individual chromosome, two chromosomes together. Bivalent or tetrad condition. Bivalent or tetrad condition. Observe carefully. Nice. Next one. Regarding the ploidy, though it is 23 pairs, number of chromosomes is 46 only because number of centromeres count it 46 only. So it is a 2n. Is there any change in the C value? No. Here 4 are there. 1, 2, 3, 4, right? Individual chromatids. Count the DNA molecules. Number of DNA molecules remains same. So it will be 4C only. So for these values there is no change, but the condition is technically getting changed. So diagrammatically you should know these differences. Clear? With this zygotin is completed. Next one is packetin. In the packetin, what exactly happens is, here also nuclear membrane breakdown. This is the centrosome moving to opposite side, right? So in this way slowly moving. Let us see. And here exactly what happens? There is exchange of chromosomal segments. What is this exchange? Some portion of this is going here and this portion is coming here. And some portion of this is coming here, this is coming here. And please remember the concept here. The concept is, generally for you at this level, we are showing that this ends only exchange. Generally you just observe 99.9% diagrams. They will show that they will use two different colors for this. This color they will add here, that color they will bring here. That is how only they will show it. but what is the original concept? At least try to understand original concept, because drawing diagram with original concept is little bit difficult. That's why they will show like this. But what is original concept? For the first one, let me draw the diagram. I am drawing like this. Don't think that it is a thread. Like, for understanding purpose, I am drawing like this. Observe carefully. Suppose this is the first one. This is the centromere. For this one, for the other one, that is also homologous chromosome only. That's why they are undergoing pairing, right? And now how exactly it will be paired is, see this. This is how it will be overlapped. And for this, the other chromatid will be like this. Understand here. In the normal diagram, at the ends only they are attaching. At the ends only they are attaching, and then they are interchanging the colors. But technically, if you see on the entire length, it is overlapped. That's why now you will get the original concept. That's why exchange of segments can occur at any location, not only at the ends. In the entire length of chromatid, exchange of chromosomal segments can occur at the entire length of these chromatids. Understood or not? And that too, this crossing over is every time a random process. That's why don't think that suppose once happened here, don't think that every time it will happen there only. It is not like this. It is a random thing. Anywhere it can happen. That is the reason why, suppose actually in the higher classes you will get more clarity. That's why in the genetics what we say it, every time you will get two chances. If crossing over occurs, if crossing over does not occur, like this, we are getting chances. Why you are getting chances? Because it is a random process. So this is how exactly the original mechanism is happening. So if you can understand this, that is well and good. Or else at least understand this. Some portion of this will go to that, and some portion of that will come and attach to this. So that's why generally they will change the colors. Okay, this is what happening in the packaging. Nice. This is what the point I wanted to tell you. I hope you understood this. Or else you just go through the normal lecture, regular lectures, so that you will get more clarity. This is the packaging. So now what you are observing here, also they are present as pairs. 23 pairs. That means total number is equal to 46. And here also still attached. That's why they are bivalent, tetrad condition. Bivalent or tetrad condition. And then the ploidy is 46 are present, so it is 2n. And the C value remains the same, no change. So it will be like 4C. Next one is regarding the diplotin. Diplotin. In the diplotin, what we are saying? Plasma membrane remains same. Nuclear membrane will undergo. Still it is undergoing the disintegration. And the centrosomes, they are keep on moving to opposite sides. They are coming like this. And here what happens? Now you will understand this. Just now I said no, in the entire length they are getting attached. Now in the diplotin what happens? Synaptonemal complex will dissolve. Wherever crossing over occur, there still they are attached. Wherever it is not occurred, they got separated. Wherever there is no crossing over, they got separated. Wherever there is a crossing over, still occurred. That's why you are seeing the X-shaped structure. Now you can understand. That's why chiasmata are present at various locations in the entire chromatid. Many chiasmata are present. So that's why here you are observing like this. But diagrammatically, how you will represent? You cannot represent. So that's why better we will draw the same diagram like packaging only. Like this only you need to draw. In this diagram, you cannot represent the chiasmata. But in the normal diagram, yes, you can represent. Suppose this is for one side, right? Like this. And here for the other one, you are representing like this. Yes or no? Now here you just observe carefully. Wherever there is no crossing over, there they have got separated. But wherever there is a crossing over, you can see this. What is the shape that you are observing? X-shaped chiasmata are observed. So in this diagram, you can understand this chiasmata concept. In the normal diagram like this, you cannot understand the concept of chiasmata. So that's why these are the original diagrams. Okay. But anyhow, but at your level, no one is teaching at that much clarity. But understand that very small concept, but very important. So in this way, you are drawing the diagram. Now what we are doing is, now we need to write this particular one. So here what we are writing, still they are attached. So bivalent tetrad condition. Bivalent tetrad condition. But what is the ploidy here? 46 are there. So it is 2n, and the C value is also 4C. This is what about the diplotin. And then here itself I will add the diakinesis. In the diakinesis, what happens? In the diakinesis, what happens is from the opposite poles. Now this. Now let us change it here. Now from this spindle fibers are coming out. And then what is the change that you need to, in this diagram no change. Anyhow, the chiasmata at in between locations, they are separated. Only the chiasmata will be attached at the terminal portion. So this is at the terminal portion they are attached. So they are present like this. So that means here also still they are present as pairs only. So bivalent tetrad 2n4c for the diakinesis. But the fully condensed chromosomes are seen in the diakinesis. Now we are going with the next one, that is metaphase 1. From now onwards, be careful. Now here, nuclear membrane disappears. So we are not drawing nuclear membrane. Suppose this is one pole, this is the opposite pole. Now please observe the previous condition. Still chromosomes are attached, still they are bivalents. If you read NCERT sentence, they said that bivalents will align. Bivalents will align here. That means still they are attached. This is one chromosome. This is the homologous chromosome. You need to attach both of these. You need to attach both of this. But in NCERT diagram, they kept us at a distance. That is wrong. That is wrong. You need to attach them. And that too, spindle fibers from the opposite poles, they are attaching like this. Yes or no? In this way, pairs are aligned here. Pairs are aligned. How many pairs? This is one pair. Like this, total 23 pairs are aligned at metaphase plate or equatorial plate. But number of chromosomes, if you see, it is 46. So here also they are still attached. So it is bivalent condition, tetrad condition. Bivalent or tetrad condition. And here what is the ploidy here? Ploidy is 2n, C-value is 4c. Understood or not? This is what about the metaphase 1. Next one is anaphase 1. In the anaphase 1, what happens? There is no splitting of centromere. Homologous chromosomes will move to opposite sides. Homologous chromosomes will move to opposite side. That means here complete dyad chromosome as it is going. Complete dyad chromosome as it is going. So in this way, on one side how many will go? 23 pairs, right? 23 top, 23 bottom. So upper 23 will go on upper side, bottom 23 will come to bottom side. Please observe carefully. In the normal mitosis anaphase, splitting of centromere occurred. Upper side 46, bottom side 46. That too they are monads. But here there is a difference. No splitting of centromere. And the dyad chromosomes are moving to opposite sides. That too 23 on each side they are going. So you need to draw all the differences. You need to draw the diagram of mitosis metaphase and meiosis I metaphase, meiosis II metaphase. Draw all the three diagrams side by side. Observe the difference. You will get more clarity. Likewise for anaphase of all the three. Telophases. Likewise side by side draw all the things. Then you will get more clarity. You can compare among these things. What are similar? What are differences? All the things with number, not just a diagram, with number also you need to observe carefully. This is what we are observing. Now what happened? Now chromosomes got individual. They got separated. Now you need to write monovalent. Once again, from now onwards, every time it is monovalent only. But here two chromatids are there. That's why it is dyad chromosome. And now for the complete cell you are counting. So 23 plus 23. 23 plus 23 is 46 chromosomes. So it is a 2n condition. And C value remains the same. In this C, 2 DNA molecules. 2 DNA molecules into 23. So 46 on upper side, 46 on bottom side. So what is the complete cell? 92. So it will be again 4C only. There is no change. So it remains same only. Understood or not? But these conditions are slowly getting changed. And with this, anaphase 1 is completed. Next one is regarding telophase 1. Telophase 1. In the telophase 1, what happens? Observe carefully. Dyad chromosomes 23 on each side. But here they will become thread-like. Dyad chromosomes 23 on each side. Thread-like dyad chromosomes 23 on each side. Nuclear membrane will be formed around this. Now observe the diagram carefully for centrosome. Now here what happens? One centrosome on this side, one centrosome on this side, right? Opposite side. This is very important from now onwards. One centrosome here, one centrosome here. Understood or not? This is the overall rough diagram for this. Now telophase means for the complete cell if you see. What are these? Monovalent dyad chromosomes. For the complete cell if you see 23 plus 23 it is 2n. 46 is 2n. And the C value will be 4C. If they are talking at one pole, if at all they are asking for one side only, now what you will write? Here also monovalent dyad chromosome. but on one side only 23 chromosomes, so it is haploid. N value, it is N, okay, it is N. Next, what is the C value here? 23 into 2, so it will be 2C. So that's why observe carefully. In general, they are asking for telophase, complete cell you need to write. If they are asking at one pole, at one side, then only this value you need to write. So read the question carefully. And then after cytokinesis 1, after cytokinesis 1, now what happens? Cytokinesis means it will divide like this. So the two cells are formed here. This is one cell, this is other cell. Each cell is having its own nucleus. Inside the nucleus, double thread like 23, double thread like 23 are present. Now not only this, centrosome, this centrosome is coming here, this centrosome is coming into this one. Understood or not? Now each cell, after cytokinesis means we need to talk each cell. In each cell you just observe, what you are observing? Monovalent and dyad chromosomes. Monovalent and dyad chromosomes. Since 23 are present, haploid, that's why at the end of meiosis 1, haploid cells are produced. But the C value is 2C. That's why in the meiosis 2, what exactly happening? Ploidy will be maintained. After meiosis 2 also N only. But after meiosis 2, instead of 2C, you will get C. That's why meiosis 2 is simply to divide the sister chromatids. Simply to divide the sister chromatids. Simply to become the DNA content half of this particular meiosis 1. This is what happening here. This is what you are writing. So in this way, actual reduction is taking place in meiosis 1 itself. Clear. Now we will go with the next one. So here what need to do? This is also undergoing meiosis 2, this is also undergoing meiosis 2. But if you are taking 2 at a time, you will be confused. So I will take 1 at a time, and at the end of meiosis 2, I will add this one also. It will be easy for us. So now we need to go with the next one. But before going to that, we need to talk about interkinesis. Why interkinesis is important? Observe carefully. After interkinesis, you need to go for meiosis 2. Again, 2 poles are required. 2 poles means 2 centrosomes are required. 2 centrosomes are required. But here each cell is having only one centrosome. One centrosome, so it should duplicate. That should duplicate. So that's why in the interkinesis, what happens? In each cell, I am taking one cell. Same will happen for other cell also. So here what happens is, this chromosome, this will be as it is only. But this centrosome, whatever is present here, it will undergo duplication. There is no DNA duplication, but there is a centrosome duplication in interkinesis. That's it. This is what happening. So that's why it will be monovalent, dyad, N, 2C. These will remain same only. Only for this one, the diagram will be changed. Clear? Now after this again, meiosis 2. In the meiosis 2, first we are going with prophase 2. In the prophase 2, what happens? It is like mitosis. So you can easily understand. Nuclear membrane will undergo breakdown. Yes or no? And here, this centrosome will move to opposite sides, and the spindle fibers will start arising from this. And here it is decondensed. But now again it is undergoing condensation. Condensation. But how many are present? 23 only are present. This is how the diagram is coming here. And now here it is monovalent. But still it is having 2 chromatids, so it is dyad. But the number of chromosomes is 23, so it is haploid. And here each one is having 2 chromatids, so it will be 2 into 23, 46 DNA molecules, so it is 2C. Understood or not? This is for the prophase 2. And now we are going with the metaphase 2. Metaphase 2. Metaphase 2. In the metaphase 2, what we are saying here? Now here there is no nuclear membrane. There is no nuclear membrane here. And here all the 23, please observe carefully. Here 23 are there, no? It is like mitosis. In the mitosis, what happens? 23 are present side by side. There is no pairing here. All the 23 like this, 23 are arranged side by side. Spindle pole, spindle pole. Spindle fibers are attached like this. All the 23 are like this. Yes or no? So here what it will be? It will be monovalent and dyad chromosome. And here it is haploid and 2C DNA amount. Next one is anaphase 2. Anaphase 2. In the anaphase 2, what happens? Like mitosis, here also splitting of centromere will occur. This is this is spindle pole, spindle pole. Splitting of centromere will occur. Here centromere will occur. So now centromere will come on both the sides. Both the daughter chromosomes are having their own centromeres. And next point is, Upper 23 will go on upper side, bottom 23 will come on bottom side. Observe the number also. Many students are not noticing the number. You need to notice the number also. Now what will you need to do? Individual chromosomes, so monovalent. But here for one chromosome, two chromatids are there. But here for one chromosome, one chromatid is there. So instead of dyad, here in the Interphase 2, what we are writing? Monad we are writing. Observe carefully. And then what is the ploidy here? 23 plus 23, 23 plus 23, it is 46. Here only 23, so haploid, but here it is becoming 46, so that's why it is diploid. Is there any change for C value? No change. 23 into 2, so 46 DNA molecule, so that's why 2C. Here 23 into 1, 23. 23 into 1, 23. So 23 plus 23, 46 DNA only. So here also it is 2C. Here also it is 2C. Understood or not? So these things compare with mitosis. What is the change? How exactly it is changed? Everything you need to observe carefully. Okay. So that's why it is about the Interphase 2. Next we need to go for the Telophase 2. Telophase 2. In the case of Telophase 2, now observe carefully. On each side, 23 chromosomes, that too monads. These are condensed, but in telophase they will become thread-like. So single thread-like 23 on one side, single thread-like 23 on other side, and a nuclear membrane around this, and here is one centrosome, here is one centrosome. Understood or not? Okay, with this telophase is complete. Now for the complete cell, if they ask, monovalent and monad, and here it is haploid, and for the complete, sorry, not haploid, diploid. For the complete cell, if you are talking, it is diploid. For the complete cell, if you are talking the 2C value, it is 2C. But at one pole, if they are specifically asking, for one side only they are asking, it is monovalent and monad. But at one side 23 are there, so it is N. And 1 into 23, so it is C value. Yes or no? That is for telophase 2. And then after cytokinesis 2. After cytokine, from this two cells are produced. But initially there is another cell also, that is also forming another. But first let us see for this one also. It is having its own nuclear membrane. Inside this single thread like 23. Single thread like structures 23 are there. It is having its own centrosome. Here for one cell. For one cell only you need to write. After cytokinesis means for each cell you need to write. For one cell if you are writing, it is monovalent and monad. It is N and C. So ultimately what you are getting? Haploid. So now these are the cells. So slowly they will mature into either sperm cells or the ova depending on the conditions. So that's why they are having that. That's why gametes are having haploid number of chromosomes, and the amount of DNA present in each gamete is called as a C value. Yes or no? This is what about the basic information. Now what is the overall summary for this meiosis 1? Meiosis 1 and meiosis 2. Initially we are starting with 2N, 2C condition. In the G1 phase initially we are starting with this. And at the end of meiosis 1, two cells are produced. And each cell is having the ploidy N. But the C value is 2C. Observe this one. And then it is undergoing meiosis 2. From this, two cells are produced, and from this, two cells are produced. Overall, how many cells? 4 cells are produced. But what is the C value? It is 1C. This is what the information you are getting. This is what the information that is related to mitosis and meiosis. And with this, we have completed all these tables and everything. Clear? This is very important concept. So though we took a little bit more time, I hope we have covered all the important informations with interlinking things. So please remember, this chapter is very important. Though it is very simple, it is very important for examination. It is having very high weightage. Okay. So with this particular chapter is completed. So before concluding this one, once again I am telling you, the videos are available on our app and also on YouTube. But the test series can be given only through the app. Clear? With this it is completed. So we can meet in the next lecture with some other chapter. Okay. Thank you so much.